All right, everybody. Who's ready? Uh-oh. I hear feedback. Hold on. Looks like I got two of me. That's even too much for me. All right, guys. How we doing? Hopefully everybody's doing good on a nice Saturday night. We get to spend the next week together. I hope you guys had a good time while I was gone. Oh, the nails. The nails are a little over the top. Are you ready for the nails? Right? The nails are a little over the top, but they're vacation nails, right? You can go a little crazy with vacation nails because nobody knows you. Exactly. Um, all right, guys, we have a ton to do. Obviously, we got microbiology. I posted to your Facebook page some, just kind of a general timeline of how we're going to be going through this. Tonight, we will get through chapter one and start chapter two. Chapter two, as I said on Facebook, is probably the longest chapter in the history of man. But it's also, of all of the chapters, the most high yield, right? So we're just going to have to grind through it and get it done. Uh, but I think we're up to it. And so that chapter will take us probably a few days to get through. Now, I did have to share because you know I was on vacation over a week ago, and I had to share the most awesome parts of the vacation. All right, so this was awesome. So this was uh, the best part of the whole thing, right? First of all, I did not get sick. And then second, I got to swim with the dolphins. So in Mexico actually. So it was all good. Anyways, my mother commented when she saw the pictures, she laughed because I closed my eyes when I kissed the dolphin. I was like, what do you want? We're in Cozumel. Cozumel. And we went to Costa Maya too. And we saw the Mayan ruins there. So it was pretty good. It's kind of like kissing a frog, right? But the dolphin didn't turn into a prince. Yeah, Deborah, you definitely got to put it. It's got to go on your bucket list for sure. Yeah, it was awesome. So it was a good time. Anyways, so I did get some kind of respiratory thing on this ship. I think it started with the little one and worked its way up. But I think we'll be able to get through it. Got a lot of cough drops. Um, got a lot of liquid, so we will we will get the job done. Okay. Um, just like before, we have the money signs. Um, and it's going to be really helpful, I think, for you because there's so many organisms, right? That's why everybody's like, oh, I hate micro. You can't hate on micro because, well, it's kind of fun. I mean, where else do we get to talk about discharges, smells, nasty sputum? I mean, this is good stuff, right? This is all why you went into medicine, so you can see worms and stuff like that, right? Anyway, so the money sign, still gonna work for you, um, and I think it'll really help you really kind of focus where you need to focus. So We'll keep that going because I think that's really helpful. The first chapter deals with some general type of information, and a lot of it though we just kind of do a little bit of an intro, but then really start to get into some of the virulence factors. So we cover them a little bit up front. That's good because USMLE Step 1, they love them some virulence factors. You know what I'm saying? They love them. So it's good. We'll focus on them in general, and then as we get to each organism, we'll focus on them for each organism. Okay. So this you can see $1 I mean, really, we had this probably in grade school, right? The difference between a bacteria and a fungi, prokaryotes, eukaryotes. Not really for step 1 of the USMLE. Okay, so the only probably thing that will help at all with in this chart really is when you get down to just looking at cell wall. So we know, of course, viruses don't have a cell wall. They can either be enveloped or not. Bacteria have the peptidoglycan, and then fungi and parasites, they have chitin, or the parasites don't have any. And then the cell membrane. If we compare mammalian cells to fungi, the fungi have that ergosterol instead of cholesterol as their major membrane sterol, and that's going to be a target for a lot of your antifungals, right? Very good. So we have to kind of prey on things that are different to kill these guys, because we definitely want to kill them, all right? Also, another target's going to be, of course, your ribosomes, right? You've got lots of drugs that target those 70S ribosomes, some that target the 30, some that target the 50. So that's also going to be important. All right. Don't want to waste too much time on that. Now, normal flora is good to kind of just get our bearings as far as the normal flora, because you're definitely going to see some of these bacteria listed in the normal flora section here as distractors at least. And sometimes, of course, they do cause disease. And we have to be aware of when probably they're just normal flora versus when they're, actually the cause of disease. And normal flora, of course, changes throughout our lives. We're born, obviously, with very little flora, and that, of course, just continues to flourish as we grow. And we know that they can cause infections when they are either lost, right? So E. coli normally lives in the GI tract, and sometimes GPS, it can't ask Siri where it's going, so it finds its way to the urinary tract and wreaks havoc, right? So that definitely causes problems there. And it can overgrow sometimes if there's some type of compromised status of the host. Now, sometimes that's induced by drugs, as in the case of C. diff, right? C. diff, that one's going to be a really important one, and that's just normal flora that overgrows when we use these broad spectrum antibiotics. And Miguel's got the other one there, bacterial vaginosis, another great example of when normal flora goes bad, right? It sounds like a B movie, when normal flora attacks. But yeah, those are the two probably, big examples for that. Now, they do contribute to our overall well-being for, lots of different areas. They are protective in the skin. They kind of create a little bit of an acidic pH so that some organisms can't grow. They help us synthesize vitamins. And then you see the competition for space. If we think about the normal intestinal flora, we really can't even fathom how many bacteria are in there, but it's just an astounding number per gram of feces. In fact, the bulk of it really is bacteria. So when you have a pathogen that tries to get in, the normal flora is like, I don't think so. We know how much space we have, we know how much food we have, and that pathogen really has to work to establish some space and room to flourish in the gut. All right. Now, we just want to take a look at some of the different body sites and some of the different organisms we may expect to see at those different sites. So, of course, our blood and tissues in general should be sterile. If we find something in the blood, it depends on which state, if we're having symptoms or not, but we could have a bacteremia, or it could progress on to sepsis. And we can have similar types of things with the fungi and the viruses as well. Cutaneous surfaces, so on the skin, right? On the skin, Staph epi. Staph epi is kind of the biggest flora of the skin. And again, there can be others, but if you put your hand on a petri dish, that's going to be what grows out mostly. Now, I want you to think, though, because we know Staph epi can cause disease, right? We know it can cause endocarditis. We know it can cause UTIs. So there are definitely areas that we can see infection with Staph epi. So we have to kind of just put that into perspective, right? If we were doing, if we had somebody that we thought was septic, or somebody that had endocarditis, and we did 3 blood cultures like we're supposed to during a febrile period, which we're supposed to, and we found Staph epi in one of the 3 blood cultures, what would that say to you about Staph epi? Right, it's not the reason. Right, exactly. It's probably a contaminant. But if you found Staph epi in 3 out of 3, either you have a really bad tech, or it could very well be what's causing the infection, right? Or even 2 out of 3, right? 2 out of 3, you're going to start maybe diving in a little deeper. Now, what other organisms can you see on the skin? Staph aureus, but we know staph likes to live right here in the big old schnoz. Got a big old nose, and probably in about 25% of the population will we find staph in the nose, right? Diphtheroids, absolutely. That's the Corynebacteria. Those are non-pathogenic Corynebacteria, so they don't make the toxin, normal flora. And yeast, we can find other species of strep. Again, but Staph epi is going to be the one you want to know. In the nares, in the nose, you got to know Staph aureus, right? That's really going to kind of be the big one for the nares. All right, oropharynx. Now, who do we want to think about in the oropharynx are mostly the viridans strep, right, which are made up of several different species of strep. So we have strep mutans, right? Strep sanguis, those are some examples of strep that are in the umbrella of the viridans category. Now, with the viridans strep, what types of things? Oh, Miguel, you're already like telepathic. Boop, 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 boop. Read my mind. Endocarditis, right? Immune compromise, Natalia, tell me more about that. Tell me more about that. For the viridans strep, they're at high risk if the patient has what? A valve. Very good. They have a damaged valve because the bacteria like the fibrin in the clot. Absolutely. Staph aureus is #1 in IV drug users, I think, is what you're getting at. And then, of course, cavities, right? Ain't nobody going to ask you about cavities on step 1 because, well, it's not the dental exam, right? All right. Other organisms in the mouth, lots of other species of strep, or the biofilms we'll talk about in a little bit. Neisseria, but here the non-pathogenic Neisseria. the non-type B serotypes of Haemophilus, and we can actually, they might ask anything. They are evil, but I'm thinking if we're putting it in high yield versus low yield, they're going to let somebody else handle the tooth decay, right? All right, then also in the mouth, but deep down in the gum tissue where the oxygen is not running aplenty, that's where we're going to find our anaerobes. And again, those are all pretty low yield. Mostly they're not going to ask you about those. The one of all of them, if they do ask, usually is actinomyces, and we will talk about actinomyces when we get to it. All right, who else do we have? We have the stomach. Not much can survive the acidity in the stomach, right? So there's no normal flora in the stomach. Who can actually live, though, in the stomach? What pathogen do you know that can live in the stomach? H. pylori, right? And H. pylori can live in the stomach simply because it has a urease. Very good, Angela, or I don't know, whoever said it. It's got a urease, so it neutralizes the acid. So that's kind of an important point. But we have tons of other bacteria and viruses and other things, but bacteria that can cause GI issues, diarrhea, gastroenteritis, things like that. And pretty much those guys just kind of shut their eyes, cross their fingers, and just are like, please, 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 let me make it to the other side, and just hope to get there and not get killed by the acidity. We have some organisms that are pretty acid resistant, like Shigella and Campylobacter, but like Salmonella and Vibrio and those, you need, and this is disgusting to think about, right? I don't like thinking about it. But you have to eat a pretty good amount of bacteria to get sick because they're pretty sensitive to the acid. In the gut, in the colon, wow, we don't even have enough time to talk about everything that's in there. But of course we know E. coli and other species actually of Escherichia. Bacteroides, as far as a genus, is one of the most common inhabitants of the gut. But Bacteroides fragilis is one of those, but it's also associated with infections. Nishat will talk about Campylobacter jejuni, causes gastroenteritis, and we'll get to that. But that one is pretty tolerant of the acid. All right, vaginal normal flora. We'd like to have a lot of lactobacillus there. Sometimes if the pH gets disturbed, if we are on antibiotics, that lactobacillus goes away, and then we wind up with the more opportunistic types of infections, either BV or, as Angela said, Candida. So yeast infections are pretty common when that vaginal flora gets disturbed. All right, good job. All right, now, you see the $5 signs here, and we started it right up front. I said USMLE loves the virulence factors, right? Because they can ask you any kind of clinical vignette. The patient presents with X, Y, and Z, most important virulence factor. So they really do focus on those, especially when we get to the toxins, boy. They really do focus on that kind of stuff. So we definitely need to focus on this, and then be able to, once we get there, associate them with the specific organisms. Okay? So, colonization. So being able to hold on, right? Seems so simple. We just got to hold on, right? Let's try to think about when that would be important. When is it important to be able to grasp onto something? You guys tell me. When would a bacteria need to hold on tight? Right? They use the pili, but Miguel's like, yep, urinary tract, absolutely. So if I'm a bacteria and I'm like, yeah, man, I'm getting out of this gut and I'm going to the urethra because I've just about had it with all, it's too crowded in here, and starts to maneuver up there. If it doesn't have ways to attach, it's going to get flushed away right by the urine. So the urine's going to flush it right out of the urethra. Now, Tatiana's like, don't flush me. All right, we won't. But what happens when we get a catheter and that flushing action is gone? What's one of the first things we see? Oh, yeah, you can say it. We all know what it is. We get UTIs, catheter associated UTIs. You got it. Absolutely. So that's going to be important. Also, let's just put ourselves in perspective. If I'm a bacteria that likes to cause, I don't know, voluminous watery diarrhea, all that peristalsis, whoosh, whoosh, I should probably be able to hold on to. So a lot of those bacteria like E-tech and some of the other bacteria that cause watery diarrhea also have adhesive factors as well. Okay? All right. So pili is typically what we think about when we're talking about adhesion, mostly for the gram negatives. Teichoic acids are the gram positives answer to those pili because those are found within the peptidoglycan layers and they kind of reach out, and that can help for attachment. And there are certain bacteria that have adhesins, and we'll talk about those because they're not as common, but we'll talk about those when we get to those specific to the organisms. All right. IgA proteases, looked up a lot of stuff about these IgA proteases, and essentially any mucosal pathogen that is a pretty good pathogen has one of these, right? Yeah, so Marcos is like, yeah, a ton of them. And if you can degrade the IgA, well, that gives you a much better chance of being able to attach to something because the IgA is not blocking your attachment, right? So that's going to be important. Now, M protein, Natalia, will get there. That has to do a little bit more with inhibiting phagocytosis. And that's going to be, that'll be important too, mostly for strep pyogenes. Okay, last but not least, biofilms. And biofilms are kind of interesting. Now, biofilms like to form on prosthetics, and essentially a biofilm, if we're talking about bacteria that form this, are bacteria that can form kind of this whole community together, right? It's like the rock concert, exactly. Miguel's got it. And I often use this example because it's a perfect example, really, of how a biofilm works. And that's the general admission concerts that I used to suffer through. Now everybody's got a seat. It's like, nobody had to like fight for your lives on the floor of a general admission concert. But in those general admission, if you're sitting in the middle of a floor that has thousands of people in it, that's right, lots of illegal stuff happens. Lots, right? And remember, if the crowd sways, you're swaying. You may not be, but you are, because they're pushing you. You have no room. And then, as Maya and Miguel said, if they're smoking weed, which everyone did at these general admission rock concerts, regardless of whether you inhaled, you still were smoking weed because you were breathing it in. And there were cops everywhere on the outskirts. There were cops by the stage. Please, they're not even going to get anywhere near you, right? Now, think of the biofilm. It's like all of the people on the floor of that general admission concert ticket or concert. They're packed in like sardines. They kind of eat together. they protect each other from the immune system because the immune system can't get in to actually attack those organisms because they're all just packed in there tight like sardines. All right, so the antibodies can't get there. If I'm a neutrophil and I'm trying to grab them, I can't because they're all tightly adherent, and then they're all kind of adherent to each other. So they're kind of protected, really. Pseudomonas is a big one for causing biofilms. The Viridan strep can cause biofilms. So those are going to be a really good mechanism for adherence if your bacteria can do that. All right, now another one that is really, really super important are going to be the capsules. Okay, $3 here only because it's a general slide, but when we get into talking about like strep pneumo, for example, you have to know that it's encapsulated, and you have to know that it's the primary virulence factor. Strep pneumo wasn't even a pathogenic organism until it got its capsule. So that's going to be important. So what you're seeing on the right in this picture, this would be a sputum gram stain from somebody that had streptococcal pneumonia. High yield for a picture. They could easily show you a picture of this, and you can see the nice little purple dots. A lot of them together, we call them diplococci. And what I want you to try to see is that surrounding those purple dots is kind of a little bit of a white halo. Hopefully you see that. That's the capsule. And the capsule does not stain with the normal gram stain reagents. So you're going to see that kind of area around these encapsulated organisms that don't stain. Now, the mnemonic that's in your book, again, I told you guys I'm not like a huge mnemonic person, but the mnemonic that's in your book is really pretty good for the encapsulated organisms. It's not by far, it's not comprehensive, but it hits all the really high yield ones. And that one is some killers have pretty nice capsules. I love the ones that relate actually to what we're talking about. So some is going to be clearly strep pneumoniae. Killers, you guys tell me, what's killers? Klebsiella, very good. Have, that one's up there already. That's Haemophilus. And who's pretty? Who's pretty? Pseudomonas is pretty. And that works, right? Because it's blue-green. It's so pretty. And then nice. Nice goes with what? And don't just tell me the genus. We need the species. Species. There you go. Meningitis. Gonorrhea doesn't have a capsule. Meningitis does. And if you get confused, that you really want to remember that, think about bacteria that cross that blood-brain barrier. Most of them have a capsule. Okay? And then Cryptococcus is a yeast. So that one not really in this section. All right, so those are the capsules, just some of the big ones. Other proteins that are anti-phagocytic. Somebody asked about the M protein. And for strep hyogenes does have a capsule, but it's not immunogenic, and it's really not anti-phagocytic. But the M protein tends to be its greatest virulence factor, okay? And the M protein is anti-phagocytic in not the way that a capsule is. And when we're talking about a capsule, it's just this kind of sugary substance that, of course, when a neutrophil tries to grab it, it can't. It just kind of slips away, right? Yeah. Yeah, it's not gonorrhea. It's meningitis, so don't let that confuse you. Okay. And now the M protein disrupts phagocytosis by inhibiting complement activation, right? Remember that complement can be an opsonin, and opsonins help with phagocytosis. So that's the main way that it tends to be antiphagocytic. The pili for Neisseria gonorrhea, antiphagocytic mostly, you know, well, there's a lot to it, I guess, but the biggest thing I want you to remember about the pili is that they change in Neisseria gonorrhea. Okay? Yeah. And then protein A, I'll show you when we get into Staph aureus pretty much how that works. But protein A actually binds to the Fc portion of an antibody, which also inhibits phagocytosis because you need that Fc portion actually for opsonization. All right, avoiding immune destruction, obviously the IgA protease that we just talked about, that's going to be important. Siderophores, all right, $1 is basically trying to steal iron, and it's not really all that high yield, so we're not going to worry so much about the siderophores. Antigenic variation, USMLE likes antigenic variation questions because this gets to how a lot of these pathogens can cause chronic infections without, essentially, it's not like we're keep getting exposed. We've got the same pathogen, and it's just being able to continue. And that's because something on the pathogen changes, and the immune system really can't keep up. The big example here is gonorrhea, where the pili change. So if you, well, think about this. When we're talking about urethritis with gonorrhea, the males are usually symptomatic, but not always. Females, a lot of the times, are asymptomatic. So if you don't know that you're infected, you're not going to get treated. And then these bacteria can cause these chronic infections like septic arthritis, right? So in a younger person who's sexually active, gonorrhea is actually going to be pretty much what you want to think about for septic arthritis in that age group. As far as the parasites, trypanosome does something similar with its outer membrane, changes the coat a little bit. That's called phase variation, but that's a pretty low yield organism in general. Gonorrhea is way more high yield, as is all the Enterobacteriaceae. And those organisms can not only change their capsular antigens, but also the flagellar antigens. Down at the bottom, you can see HIV. And the other one I would probably add into that is going to be influenza, right? Nishat, yeah, absolutely. Because those are both really high yield and both really medically important. So antigenic drift. And, if you think about both of those viruses, they're both RNA viruses. And the reason that those viruses drift is because the RNA polymerase, also the reverse transcriptase for HIV, doesn't have a proofreading function. So lots of mutations happen, and they don't get fixed as the DNA polymerase would, generally fix them. So shift is something completely different. Not really considered antigenic variation, but it's a complete change in the phenotype of the virus. So when we talk about influenza, we'll absolutely talk about shift, but that doesn't really happen the way that this does. This explains kind of, why the immune system can't necessarily keep up, but a shift is something different, and we'll talk about that a little bit later when we get into the viruses. Okay, intracellular survival, invasions not very important, but the organisms that are facultative intracellular, and tuberculosis is the most high yield, but still there are others, and we'll talk about them as we go through the bug parade. But those are going to be really very high yield. So whenever you hear the word facultative, I want you to think that the bacteria has a choice. So in a facultative intracellular pathogen, those bacteria can live inside of a macrophage, or they can live outside. They like living inside because they're protected from the immune system. Mincho says granuloma reaction, absolutely, and we talked about this a lot with immunology. We need that granuloma reaction because they're escaping, right? In some way, shape, or form, they are not getting killed within those phagocytes. So those are going to be high yield again, and we'll talk about those as we go through. Invasins, not really super high yield. When we talk about facultative intracellulars, we're talking about bacteria that get into phagocytes that then escape killing in the phagocyte. Invasins are proteins that some bacteria have that can allow a non-phagocytic cell to take it up, which seems like a brilliant idea, right? Because then you don't even have to worry about killing, and you're still kind of hidden from the immune system. You're correct, Miguel, that Legionella is also facultative intracellular. There are some others too that we'll go through, but you definitely want to look at those. Type 3 secretions, nope, not high yield at all. This is just a way that, you know, if I'm a bacteria and I make a toxin, that I can deliver that toxin directly to a host cell. So we usually kind of, compare it to a syringe. So it's like a little molecular syringe, and it just injects the toxin directly into the host cell. A lot of the times, the toxins can be detrimental to the bacteria. So they just want to get rid of it, and they can target it right to those host cells. So yeah, it's pretty cool, just not very high yield. Jet motility we'll talk about, because that's going to be important for a couple of bacteria, and so we'll definitely talk about that, especially when we get into listeria. All right, hopefully you guys, I know you know this, because I know we spent 3 days together talking about immunology, and we know some of the different immunologic mechanisms, right, that can cause damage to the host. So we know our type 2 hypersensitivities, that's rheumatic fever. Our type 4s, that's the delayed type. Our type 3s, that's our immune complexes, right? And post-streptococcal glomerulonephritis is a good example. Hepatitis B, another good example of when we get immune complexes. And then the last one down there, this one's kind of an interesting one. In general, and I think we mentioned this when we were doing the immunology, but peptidoglycan itself and teichoic acid are both chemotactic and will recruit neutrophils if present, right? So if we have bacteria, now normally when we're talking chemotaxis, we're talking about inflammation. But when those neutrophils come, they're going to be looking for something to kill. Now, one of the, I don't know that it's a trickier question, but USMLE question kind of regarding this concept is something like this. So you have a patient, they'll kind of build up the vignette. You've got somebody with strep pneumo, so they've got fever, shaking chills. They have the rust-colored sputum. Maybe they give you the fact that it's a sputum gram stain, and you can see the neutrophils in there and the organisms. Now, for strep pneumo, we know that it's encapsulated, and so the neutrophils cannot phagocytose those organisms because of the capsule. And so the question is actually, then why are the neutrophils there, or what got the neutrophils there, right? Because they're not capable of phagocytosing. Well, it's the peptidoglycan, it's the teichoic acid. It's, other things in the inflammatory response that are recruiting those neutrophils there, even though they can't actually clear up the infection. Okay, so it's all about recruitment for that, and that's the peptoglycans, anticoic acids are both chemotactic. Now, physical damage, we'll get to this mostly when we talk parasites, because everything else is really pretty small. Ascaris will be by far the worst thing that we talk about together, because it's the largest thing that we can be infected with, and it can sometimes cause blockages because those worms are really so big. Entamoeba histolytica can be very aggressive, and once it gets released, it can cause ulceration, can release bacteria, but it can also cause some damage to the liver as well. All right. Now, toxins, lots of money on the toxins, all right? And they know that you get confused sometimes with terminology. And we said they were evil. Absolutely, they are. And they're going to use the terms that confuse you on purpose, right? Doesn't that sound horrible? It kind of goes against every educational philosophy that we learn. And then there's just the general be nice to each other that seems to go out the window here. But you need to, without a doubt, know that if they're using the term endo, kind of meaning within, endotoxin, they're talking about LPS. All right? So you're absolutely right. If we're saying endotoxin, we're talking about LPS, lipopolysaccharide. Where do we find LPS? Angela read my mind. We find them on gram-negative bacteria. Sometimes you'll mistakenly hear that Listeria has LPS. It does not. First of all, Listeria is gram-positive. And Listeria has lipoteichoic acid, but it does not have lipopolysaccharide. Okay? So it was mistakenly thought that it may have been LPS, but it's not. So there's no gram-positives with LPS, Miguel. So that's what I'm trying to say. You may have heard that Listeria does, but it does not. So the gram-negatives are the only bacteria that have the LPS. Now, you do have to know for LPS that it's the lipid A portion of the LPS that's toxic. That's the toxic part of the molecule. Everybody else is polysaccharide. So it's the lipid A that's toxic. Now, LPS, we know, is a very potent mediator of the immune response, right? We know it can bind directly to receptors on phagocytes, causing them to become activated, and they'll release cytokines, and we can actually send a patient into septic shock or disseminated intravascular coagulation, or DIC, disseminated intravascular coagulation. You'll have I'll tell you where the questions come in for LPS, and where it is going to be the most important virulence factor is in a case of sepsis or septic shock. So let's say you have a patient, usually they're hospitalized, they got some kind of contamination maybe in the central line, and all of a sudden they start getting fever, chills, and then some of the symptoms of shock, right? Blood pressure dropping, all kinds of stuff like that. And they'll tell you, they don't necessarily tell you what organism it is, but they'll say gram-negative rods were isolated from the blood or from the blood cultures. And then they're going to ask you which virulence factor is important for those symptoms. And you might be saying, well, I don't know what organism it is, so how can I answer this question? All you need to know is that they're gram negatives, right? And gram-negatives have the LPS, and it's the LPS that causes those symptoms. So that is 100% where you'll get a question on the endotoxin, or LPS being the primary virulence factor, right? So that'll be important. Now, what's probably even more important, though, are the exotoxins, because they love to hit you on the exotoxins. And the exotoxins have lots of different functions, and we'll talk about what those are. And you're right, it could be the exotoxins. It can be found in gram-positives and gram-negatives. So you could have a gram-negative with exotoxin and endotoxin, okay? E. coli is a good example. The exotoxins are secreted, right? And they're secreted, and they essentially have a classic kind of function where we call them AB toxins. And it's really easy to remember because B binds, so the B part of the toxin binds to the host cell receptor, and the A part of the toxin is the active part. That's the part that's going to kick your butt, right? So that's the part that's going to give you some type of change of cellular function. Okay, and we'll talk about what some of those are. The cytolysins in general are much lower yield than the neurotoxins and enterotoxins and things like that. But the one that is high yield is going to be the alpha toxin from Clostridium perfringens, because that toxin is directly responsible for causing myonecrosis or gangrene. But mostly we want to be able to focus on the AB toxins. All right, now these toxins are kind of broken down into their function here, and again we'll talk about them when we get to the specific organisms. But in general, we have toxins that inhibit protein synthesis, and they can do that one of two ways. They can either interfere with the ribosome, or they can interfere with elongation, right? ADP ribosylation of elongation factor, inhibiting protein synthesis. The way that I remember these is I remember that Shigella is the one that affects the ribosome. Yeah, so I put the money on the beginning slide, the one that we were just on. So these are all $5 sign, right? But I remember that Shigella is the one that interferes with the ribosomes. And then if you remember that, you should remember that EHEC has a Shiga-like toxin, right? So you got Shigella, Shiga-like toxin. Those are the ones that interfere with the ribosome. Then you just have to remember the other two are the ones that interfere with elongation. So Pseudomonas and Corynebacteria. So that's how I remember those. And you see here the EHEC toxin, and we'll get into that. The neurotoxins, obviously, those are going to be pretty high yield, and those are all Clostridia. So those are Clostridia phenomenon, tetanus and botulism, and they kind of function a little bit backwards from each other, right? One causes a flaccid paralysis, and the other causes like a spastic paralysis. And we'll get into those when we talk about the different types of Clostridia. Super antigens, we talked about in immuno. We know that they're super high yield. We know you're going to get a question on super antigens. EHEC is enterohemorrhagic E. coli. We'll talk about it probably, I don't know if we'll get to it tomorrow, but for sure on Monday. And we're going to need some work when we're talking about EHEC, because it causes a bloody diarrhea, but it's not the same as all the other things that cause the bloody diarrhea. All right, now on boards, and I think we said this during immuno, if you get a super antigen question, they're not going to be asking you about strep pyogenes. They're going to be asking you about toxic shock syndrome toxin, which we know overstimulates the immune system because it binds to that T cell receptor outside of the normal peptide binding groove. So instead of getting a very specific immune response, we get, that's right, Nishad, a cytokine storm. Absolutely. Super Hulk. Bad Hulk. Very bad Hulk. All right, then we have the ones that increase cyclic AMP, the cyclic AMP inducing toxins. And just as a generalization, they really, when we're talking about these toxins, we're releasing water from a cell. So we're getting edema, we're getting diarrhea, whatever organism we're talking about, you just want to kind of think about what the toxin does and kind of relate it back to that function. And again, mechanism is going to be really important, so they will ask you that. I know biochem talks a lot about the different toxins because they can show how things interfere with function and things like that. And we'll get into talking a little bit more about them when we get to the organisms that they're specific for. The cytolysins, remember I said, are not that important except for Clostridium perfringens. So this one, I mean, it's not as high yield as the rest, but I may give it like $2 or $3 signs versus the alpha toxin for Staph aureus. When you got toxic shock syndrome, the alpha toxin just isn't going to be as important. Okay? All right. All right, guys. Well, I think we're at a perfect stopping point. So we'll go ahead and take 10 minutes here, and then we'll come back. We got a little bit more intro stuff that's in Chapter 2, and then we will just dive right in to talking about some bacteria. All right? So I will see you guys in about 10 minutes. So I know what you guys do on break. You guys eat, and then you don't share. We will never get through all of Chapter 2. Absolutely not. So we're just going to start it and see how we do. Abdullah's getting pizza. I know. Oh, Haley, no. All right, there is something that does not like to work when I try to put that camera back on. I don't know what the deal is, but once I shut the camera off, it's like, yeah, no, you're not allowed to go back on there again. It's totally the gremlins, and it happens pretty much every time. All right, guys. So hopefully you had a good break. You walked around a little. We're going to start chapter 2, which don't get too excited because there's still some intro stuff in here we have to cover. But it eventually means we'll start talking about some of the specific bugs. Adam's not the gremlin. Adam makes it all work. It's all black magic, right? All right. Bacterial cell walls and cell envelope and stuff like that. You can see there's $2 here. You really just want to have a general idea about these. And the biggest differences, of course, are going to be the peptidoglycan. The peptidoglycan is much thicker in the gram-positives. Gram-negatives also have peptidoglycan, but it's much thinner. Very good. And then, of course, the gram-negatives have the outer membrane, which the gram-positives do not have. And it's that outer membrane where you're going to find the LPS. You got it. So the LPS is going to be housed in the outer membrane of the gram-negative bacteria. And because now in a gram-negative, you've got a cytoplasmic membrane and an outer membrane, you've got that space in the middle. It's called a periplasmic space. And if you flip back over here to the gram-positive side, you can just see some of the teichoic acids. We said that those were important for attachment, but also chemotaxis, right? They'll recruit neutrophils. And you can also see, sorry, Haley, I'll keep my nails underneath the screen. The lipoteichoic acid, which remember we said got some people confused that Listeria may have had LPS, but we know it doesn't. All right. Questions that can arise from this type of stuff really mostly just deal with simple things like, let's just say peptidoglycan, for example. If I asked you about a gram-positive organism and what feature protected those bacteria from lysing easily, it's the peptidoglycan, right? Because it's really thick. Just kind of things like that. They're going to be few and far between. They're not super high yield, but again, they could come up. They used to on USMLE before it really got converted over to lots of the clinical vignettes. They used to just have diagrams of these, and you'd have to kind of tell which each part was. The gram stain, again, really they're not going to ask you if they're not really going to ask you about the gram stain per se, as far as what's going on, but it is kind of a good idea to know that the gram-positive stain purple because that crystal violet stays in the peptidoglycan. All right, so you've got a lot of tables in there that I'm not going to go over because it pretty much just talks about each part of the bacteria and gives you function, things like that. Again, not super high yield. Endospores, also not super high yield. You do need to know who makes them. That's going to be important. That's high yield. So Bacillus and Clostridium. We want to know that spores are a survival mechanism. And, sometimes, wouldn't you guys all like to be a spore sometimes? I think sometimes that I would. Like when stuff gets really stressed at work, that you could just form a spore and then let the stress go away and then come back when it's all, or in the winter. We could all sporulate in the winter. And then in the spring when the weather's nicer, boom, we just come right back out and start to party. I know. It's freezing here too. It was, I think it was a high of 19 today. right? Or hibernate, yeah, but then I don't know about hibernation, because then you just pretty much sleep the whole time. I don't know. I would just like to turn into something else, I think. But anyways, these spores are very, very resistant to chemicals. They're resistant to heat. They're resistant to drying, freezing, all kinds of things, because they have several layers of protection. But inside the core is still the DNA of the organism. So the way that these spores work, I just want you to think about for a minute, because it will be important when we start talking about some of the organisms that make them. If we come in contact as a host with a spore and not a bacteria, can we get sick from that spore? Yes, we can, right? Because now the spore is in a nice warm environment, and you got lots of food for it to eat. And so each one of those spores that come in contact with you will turn into what we say a vegetative bacterial cell, which means one that can now replicate. So one spore turns into one bacteria, and then that bacteria will replicate. So as far as mechanisms of resistance, things like that, you really don't need to get into that. You need to know Bacillus and Clostridium, and you need to know essentially that the spores are very resistant in general. Okay. All right. Now this is going to be taxing, so I do hope that you're ready for this. This is how bacteria replicate. Okay. All right. Try to stay with me. It's hard. All right. One bacteria makes two, and two bacteria, they split off and they make four. It's mind blowing. And then four make eight. I'm already exhausted, mentally exhausted thinking about it. It's just binary fission. They divide, boom, they're each a copy of each other. Now, the annoying part about binary fission for you and I is going to be that if we replicate by binary fission, and one of us makes two and we're an exact copy of each other, that's pretty boring, right? And there's not a whole lot of room for change, i.e. mutations, and it's all in all pretty dull. Minus some random mutations that happen, how would we ever expect to get better? Yeah, not very easy. So the annoying part about binary fission for us is that we have to talk about genetics. Yeah, and that's not fun at all, right? So that is how bacteria acquire new genetic properties, because they replicate like this so boringly. One makes two, two make four, and now we got to talk about all those different mechanisms of genetics, right? So we'll talk about that in a later chapter. We won't ruin tonight by even thinking that we have to talk about genetics. No, because those are some pretty dull chapters, I'm not going to lie. All right, you don't have to know, that's pretty much it for, I know, it's Saturday night. We're not going to ruin it. All right, bacterial growth curve, again, you see $1 sign here. They're not going to ask you much about this growth curve. Again, it's not super complicated. In the lag stage of bacterial growth, they're just kind of gearing up to replicate, like they're getting all their stuff together, right? Making the enzymes they need, just getting ready. But once they get ready, they kind of hit the ground running. Like E. coli, ain't nobody going to replicate fast like E. coli. And so that's when you enter that logarithmic stage of growth. And then, well, things start to happen, cells start to die, and we've got some cells dividing, some cells dying, and they about to equal out. So that's when we get to stationary, and then, well, everybody's got to go. So then the death phase, we got more cells dying than are replicating. Again, not too taxing, not a lot of questions that can come from this. About the only thing that they could really probably clinically ask you about this is at which stage of the bacterial growth curve are antibiotics the most basically, do they work the best? Are they most effective? Yeah, it's going to be the log phase. When they're actively replicating, we're just going to beat them down with drugs, right? If they're already dying, they're not going to be, because they're dead, right? Right. So it's that logarithmic stage, but that's really it. So nothing high yield here. We're not going to focus on that. I know there's some questions in there about calculating the number of bacteria. Really, nobody's going to make you do that. So that's kind of old school stuff, and they're not going to make you calculate the number of bacteria in a growth curve, right? All right, so we're not gonna worry about that. All right, culturing of organisms. So here's my take on culture. You're not going to get a question that says, "Hey guys, what grows on buffered charcoal yeast extract?" Right? I know. I know. I was sweating that out too. But you're just not going to get it. Now, here's where this can become somewhat important. If I'm getting a vignette and, it's describing, I've got maybe a 60-year-old male, they got kind of a dry cough, they're a smoker, they were at a Legionnaires convention, you know the drill. And I say that we isolated the organism on buffered charcoal yeast, you may already know what it is, and the buffered charcoal yeast just kind of seals the deal, right? It's like, oh yeah, okay, yeah, I know that's Legionella. So again, it's not going to be something that they ask you, but in a vignette can be very helpful. All right. So there's a few that we want to talk about, and make sure that we know. Obligate intracellular. But in a question about an obligate intracellular organism, they're basically going to say, that we found these intracellular inclusions, or, if it's viruses or something, they'll give you a clue like that. As far as the lab media, we've got lab media that can be selective, all right? So some examples of a selective media. Well, first of all, what is selective? We're selecting for the growth of certain organisms by inhibiting the growth of others. And Thayer-Martin agar is a good example of a selective media, right? Because we're selecting for pathogenic Neisseria. Now, a differential agar helps you, just what the name suggests, differentiate between different bacteria based on some of their properties, right? And the best one we can probably use is McConkey, right? Nishath got it, McConkey, where we can differentiate between bacteria that ferment lactose and those that don't. But I will tell you, and this is a nice big clue about McConkey, so you want to write this down, because this is going to be important. McConkey is also selective. And we don't often think about that, but only gram-negative rods are going to grow on McConkey. Gram-positives won't grow, okay? So what that means for you in a question is this. I'm reading a vignette. It says blah, 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 something grew on McConkey. And I look in the distractors, and I see some gram-positives. You know what that means? That means we can cross those right off the list, because gram-positives don't grow on McConkey. So that's where these agars, just by knowing a little bit about some of the really more important ones, like McConkey, are going to be a little bit helpful, okay? Now, yeah, lactose fermenters will make it pink, absolutely. So I'm going to go through, there's some that are highlighted here as important ones. And I agree with some. I don't necessarily agree with the others. Anaerobes, they're not going to talk to you about culturing anaerobes. They're just going to tell you that it did not grow in the presence of oxygen. That's going to kind of be your big clue. Corynebacterium is high yield for boards, but they're probably not going to talk about culturing it necessarily. McConkey's is a big one, so I would highlight that one. They're going to ask you about McConkey. Vibrio used to be really high yield. It's not so much anymore. So the fact that it grows on TCBS, not super important. Now Legionella grows on the buffer charcoal yeast that we said. And, that's important. Legionella is one of the atypicals. You got about a 50-50 chance of getting a Legionella question. So just to know that it grows on the buffer charcoal yeast, yeah, that's probably pretty important. Mycobacterium, again, probably not going to talk to you about what it grows on because it takes so long to grow. So for something like TB, you're going to have to get it based on the clinical vignettes. Now, what is important are the last two down here. So chocolate agar, we definitely need to know what grows on chocolate agar. And then, of course, we already mentioned Thayer Martin. That's going to be important. That is chocolate agar with antibiotics in it to suppress the growth of normal flora, but it grows the pathogenic Neisseria. All right. Chocolate agar in general is an interesting type of agar because we will talk a lot when we get to the gram-positives about blood agar. And we'll talk about alpha, beta, gamma, hemolysis, all that good stuff. But chocolate agar is just blood that's been gently heated so that the blood is all lysed. And we need that to grow organisms that don't have hemolysin, anything to lyse the red cells, right? Because if you can lyse red blood cells and you actually need those nutrients inside the red cells, is no problem because you lyse the cells and then all the nutrients are there. But chocolate, you want to think Haemophilus because those are all going to be pretty much grown on chocolate agar. And then Neisseria, again, probably if they're talking about Neisseria, they're going to talk about the Thayer Martin agar. Okay. Now, I will just take a couple minutes and tell you a funny story about Thayer Martin so you'll never forget what grows on it. Is that when I first started teaching, I taught, and this is probably 15 years ago, okay? I was teaching with one of my best friends from school. And there was this kid in class that we really liked. He was super smart, but he was always kind of like the smart aleck too, which I'm good with. If you're a smart aleck, we're going to be best friends, okay? So we did the lab, of course. You've all probably done this where you got to culture your own throat flora. And when all the students left afterwards, we tainted his Thayer Martin plate with gonorrhea. Totally thought, okay, we'll play a good trick on him. So we let them culture on blood agar and Thayer Martin, just kind of as a joke, right? So we come in the next day, and the girl sitting next to this kid was crying. Oh yeah, she was crying. So we kind of snickered, like, ooh, they're probably dating. Yeah, no. He, because, of course, he's going to cause me problems, he wrote his name on the plate from her throat because he cultured it, right? That makes perfect sense. So he wrote his name on her throat culture. So we basically tainted her culture with gonorrhea. Yeah, that was really bad. And then after that, we thought we'd probably get sued if we ever tried that again. So we stopped doing that. But anyways, I know you got to have fun, right? You with me? Yeah, you got to have fun. Now, tainting someone's plate with gonorrhea may not be your kind of fun, but it's my kind of fun. Microbiologist, what can you say? I know. So anyways, that'll help remember that Thayer Martin grows gonorrhea. All right. Next, different types of growth requirements for mycoplasma, cholesterol, purines, pyrimidine. You got to add that in the media. That's not really high yield. Cysteine. Now this one is easy, so I would remember probably this one, but all of the Ellas, like Francisella, Brucella, Legionella, Pasteurella, all require cysteine. So they can get that off the buffered charcoal yeast. So some of those can grow on that. So right, all of the Ellas. And then of course another big one, and you guys all know this, are X and V. So Haemophilus requires X and V, heme and NAD, and that's within the blood cells, and so we need the chocolate agar to get it out of the blood cells so that the bacteria can access those. All right. Now, oxygen requirements. Not important. So you got $1 here. But I just, let me qualify that for you. So are they going to say, hey, which bacteria are obligate aerobes? No. But it's kind of sort of like the media. All right? They'll put it in the question, and it'll be kind of a clue to you if you know the bacteria that kind of fall under that category. So let's, for example, all right, let's take obligate aerobes. So these are bacteria that can't ferment. They require oxygen, and there are several probably really good examples here. But the one that I like to use is Pseudomonas. We'll talk about this one first. So Pseudomonas is an obligate aerobe. Now, think about when we talk generally about Pseudomonas, it's a gram-negative rod. We talk about them together with all of the enteric bacteria, right? So E. coli, Salmonella, Shigella, because it can cause similar diseases. Now, one of the clues that they may give you in a vignette, if they're looking for you to pick Pseudomonas, is that we found gram-negative rods wherever, okay, that were non-fermenters, okay? Gram-negative rods were isolated from pick a body site that were non-fermenters. That's a pretty big clue because most of the gram-negative rods we know are fit under the enteric category. And so that's going to be a big clue that maybe you have Pseudomonas. Now, Pseudomonas has lots of other clues, but that could be one as far as oxygen. Mycobacterium, tuberculosis, grows in the apex of the lungs. Absolutely 100% requires oxygen. When I teach these classes, a lot of the times when we're doing them live, I'll get all kinds of questions and comments during the breaks and stuff. And I had one student, I can't remember exactly where she's from, but she asked me, well, she basically said where she lives, they bring patients that have tuberculosis to the highest elevation point in the village, and that this seems to be pretty effective. And I was like, that's pretty cool, right? So you're taking advantage. Oh, so it's done in Nepal. So taking advantage of the fact that the bacteria can't live in reduced oxygen. So the higher up you go, the less oxygen. Tuberculosis can't do as well. So those are all just kind of simple examples. Microaerophils, those are organisms that like some oxygen, but they also like some CO2. And again, that's really not going to be anything that they ask you about. The majority of bacteria are actually facultative anaerobes. And so that's kind of cool. Remember what we said about the word facultative. Means we have a choice. And the same goes when we're talking about facultative anaerobes and we're talking about facultative intracellulars. They're going to use oxygen if it's there because they have a choice. And then when the oxygen is gone, it's like, no probs. I'm just going to ferment, right? So think about our gram-negative, all the Enterobacteriaceae, right? And think about when we have anaerobic infections, and a lot of the times they're abscesses, and they're oftentimes mixed infections, right? Most of the time polymicrobial, and they stink, right? That's one thing that you'll definitely see with anaerobic infections is the smell, and they smell because of all that fermentation that's happening. And the organisms that will cause those polymicrobial infections with the anaerobes, they're going to be the enterics because they're going to use the oxygen if it's there, and if it isn't, no biggie. We're just start to ferment. All right. Obligate anaerobes, three major ones that are tested on USMLE: Actinomyces, Bacteroides, Clostridium. So remember the ABCs of anaerobes, and that'll pretty much get you through anything that's anaerobic for the most part. Of those three, Clostridium is going to be the most high yield, obviously. And what they will put sometimes in the question to let you know that it's an anaerobe is that the organism would not grow in the presence of oxygen. So they'll say, we isolated the bacteria and it did not grow in the presence of oxygen. Well, that's a pretty big clue. Okay? So again, that's just kind of the level. Haley, I'm jealous. That's just kind of the level that we need to know the different oxygen requirements. So really not that big of a deal. All right, gram stains. You do not have to know the procedure, but listen to me and pay attention here. You have to know colors, right? So they're going to put up a picture of some purple round bacteria, and you're going to have to know that's a gram-positive cocci. Or they're going to put up a picture of a pink rod, and you're going to have to know those are gram-negative rods. So colors undoubtedly are going to be important. I really don't know what they do for colorblind people on this test, because this is a problem. You have to be able to differentiate colors. And in almost every class I teach, there's somebody that's colorblind, right? And so it's a little more challenging for them. But you will have to interpret gram stains, but you won't have to go through the procedure. Okay. Same holds true for the acid-fast stain. You don't have to know how to do it, but you have to know how to interpret one. And so the big clue is that anything that's acid-fast is going to be pink. So it's going to stain this hot pink. And anything that is non-acid-fast is going to be blue. So, for example, in tuberculosis, if I did an acid-fast stain, if they tell you they did an acid-fast stain, but they don't tell you the results, because, that's nice, make you interpret the stupid thing on your own, and they put up a little, acid-fast stain. If you see pink rods in an acid-fast stain, those are acid-fast bacilli and probably tuberculosis. So, you again, want to be able to interpret it, don't have to be able to go through the procedure. And they definitely have those up there. Okay. Now, this is kind of the last of the intro stuff before we get into really the massive bug parade that we're about to embark on. But I'm gonna just take some time to talk about how important this is gonna be, okay? Now, I'm fully aware that everybody's got different favorites as far as subjects and things, and maybe micro's not your thing. Maybe micro sucked where you went to school. I don't know. But because sometimes it does. Sometimes maybe you just have this much micro and you needed this much. All different types of things, right? So people have different levels of comfort as far as the gram reactions and the shape. So this is super important. Yeah, you do. You do have to memorize it, but not like this, all right? Let's do something. Why would your teacher hate you? That's horrible. Let me guess. I'll give you the number one reason that teachers hate medical students. You ready? Because they tried to get into medical school themselves and got denied. Exactly. That's why. Okay, maybe youth, but I said number one reason, right? I can't tell you how many of those we had. They're all the people that are mean to students are always the one with an axe to grind, right? They're jealous. So you don't let any of that stuff bother you, right? Okay. So back to business here, okay? You have to be comfortable with the gram reaction and shape of all these bacteria. And there's an easy way to do it. So you guys, I want you, we'll interact together a little bit here, but there's 4 general categories, right? We have gram-positive cocci, gram-positive rods, gram-negative cocci, and gram-negative rods, right? I mean, in general. And then under gram-negative rods, you've got some that are curly and other things like that, but those are your 4 general categories. All right, now, tell me who are the gram-positive cocci? Staph and strep. Yep. Well, that one's pretty easy, right? So if they say we found gram-positive cocci in the pick your body site, we're going to know it's staph strep, or as some of you guys said, enterococci. You can't forget about the enterococcus. We like to, and it starts with entero, which even in my brain sometimes wants to put it in the gram-negative category, right? But it's not. It's gram-positive, all right? So staph, strep, and enterococcus for gram-positive cocci. All right, gram-positive rods. It's a bigger list, but it's still short, right? It's still short. So we have bacillus, listeria, corynebacteria, and what else? There's one missing. Oh, Clostridium, right? Clostridium. Those are the big 4 of gram-positive rods. Bacillus, Clostridium, Listeria, Corynebacterium. Okay? Then we have our atypical gram-positive rods, and that's Actinomyces and Nocardia. So those kind of branch off. They look like, kind of like fungi, and I'll show you some pictures. But all in all, again, you memorize more than that the first day of anatomy, I'm sure. So that's not bad. Yeah. I know you guys have to come up with the mnemonics, because it's harder for me to do that, because I don't know, I've been doing it so long, they just like, we know them, right? So you guys will have to come up with them, and then we can share them with everybody. All right, gram-negative cocci, hit me. Go on, hit me. Gram-negative cocci. Yeah. Neisseria is going to be the most high yield, and then Moraxella, which, bam, probably they're not going to ask us about. Okay, so that was pretty easy so far, right? You guys are like, mnemonics? Hell, we don't need mnemonics. This is easy. All right, now here's the important point. So I'm going to get real close to the camera. Everything else is a gram-negative rod. Oh, not so bad, right? Everything else is a gram-negative rod. Now, just like us, we're all humans. We're all mammals. We look a little different, right? We do. We look a little different. And now we just have to know what some of those differences are. Okay? Are we curved? Are we spiral? Do we grow at a certain weird temperature? Do we ferment something that we can pick up on? So it only really starts to get tough when we get into the gram-negative rods, and that's why we have to go through all the flow charts and all that important stuff. Okay, now I'm going to type some stuff up here while I'm chatting with you, and we're gonna talk now. It's definitely not always as easy as I'm gonna paint it right here, but I'm essentially gonna tell you why this is important. All right, so we got one, two. I'm just drawing some bugs up here. Okay, almost done. And okay, now it's not pretty, I'll give you that. Okay, here's a list of bugs. So here's the way these are going to work. You want some toy bugs? I know. Sometimes when I go live, I bring them and give them out as prizes, but that's hard to do now, right? I know. It's hard. Okay. Now, here's a list of organisms. Here's why this is important. And here's the way the vignettes are going to go. Sometimes gram reaction and shape doesn't mean anything, because they're going to tell you this. They're going to say you have a patient, and they say it's a neonatal meningitis case, right? So you've got a neonate, they've got a bulging fontanelle fever, maybe projectile vomiting, okay? And they're just going to say, what's the most likely cause? Okay, so can you pick out for me on that list, what is the most likely cause of neonatal meningitis? Right? Strep A galactiae or group B strep. Yep. Okay. So in that case, gram reaction and shape and everything I just told you is crap. But that's not going to be mostly what they do. Okay? So mostly they're going to give you the symptoms. All right? So here I am, again, neonatal meningitis. And I say that gram-negative rods were isolated from the CSF. All right. Now, knowing the gram reaction, and actually, if you look at this, the gram reaction is the only thing you needed to know. Because the only gram-negative rod in that whole list is E. coli. Right? Now, here's where kind of knowing what bug to expect in what disease and what age, you know, which ones are going to be the most important, is something like this. So maybe I give you that same case and I say gram-positive cocci were isolated from the CSF. All right? And you say, okay, so just based on that, I can cross off Neisseria, E. coli, and Listeria. That leaves me with Strep A galactiae and Strep pneumo in this list. And now I know what's most likely, so I'm going to pick Strep A galactiae. Okay? But knowing that gram reaction and shape, and just knowing that as a good foundation for getting started with some of this, I can't stress it enough. Because you will be able to cross off certain answer choices just based on them telling you what the gram reaction and shape is. Right? So it's going to be really helpful. And my point and why we're spending some time on it is that if your foundation is shaky on this part of micro, it is not a waste of time for you to spend some time going through these and making sure that you know these gram reactions. Okay? Make flashcards, quiz each other on Skype, whatever you want to do, but you know how to build a house, right? And it starts with a firm foundation, and then you build from there. The house is going to be a little shaky if the foundation isn't solid. Foundation, and then everything else will kind of build off of that, right? So again, gram-positive cocci, staph and strep, and enterococcus. Gram-negative cocci, Maria just kindly put up for us Neisseria and Moraxella. By far, Neisseria is the most important. Gram-positive rods, Listeria, Corynebacterium, Bacillus, and Clostridium. And then we have Actinomyces and Nocardia. And then everything else is a gram-negative rod. Okay? Everything else. All right? So when we get to the gram-negative rods, what are we going to focus on? We're going to focus on how on earth are we going to differentiate them one from another. Okay? Yeah, micro is really pretty high yield. Is job security. I'm okay with that. Actinomyces is a gram-positive rod but it branches. It has little branches on it. So when we get there, I'll show you some pictures of actinomyces. Okay? All right. So did I make my case? Are you guys going to go back and look at this stuff? Hopefully. Yes. All right. Woo! Score. All right. Good job. So let's. Oh, that was going to be where I drew some of that stuff, but my drawing's not very good. All right. So gram-positive cocci. You told me staph and strep, and then enterococcus. Now, enterococcus actually used to be a strep, which somebody asked, why isn't enterococcus listed? That's why, because it used to be a strep. So it kind of really is fitting under that category. Now, staph and strep, they're both gram-positive cocci, can cause similar diseases. And here's a question they love to ask, and I know some of you are already thinking it because you're telepathic with my brain. But if I have a patient that is impetigo, what's the simplest way for me to tell what organism it is? You got it. Catalase test. 100% correct. Now that's important. That's high yield. They absolutely will ask you something about that silly catalase test. So that's a nice way we can differentiate staph from strep. Can we ever clinically differentiate staph from strep just based on the gram stain? Because, you know, strep's in chains, staph's in clusters, yo. Absolutely not. No. I've seen so many gram stains of strep that look like staph because they're thick. It's not enough. So catalase. So who's catalase positive? Staph is catalase positive. And then, of course, we know there are other organisms that are catalase positive, and we'll talk about those as we get through. And now don't hurt my feelings here, because I'll start to cry. But what disease is an immunodeficiency disease where we see recurrent catalase positive bacteria? I'm afraid to look. Nice. Woohoo! Chronic granulomatous disease. Nice. Or Natalia's just like, I'm going to one up, and it's not just CGD, it's the NADPH oxidase deficiency. Absolutely. Because both of those, that's going to be super important. So again, just kind of bringing it back to the immuno, all of that's going to be really high yield. But here's where we get into it. Now, Deborah, if I didn't make this fun, we'd all be sleeping, right? But thank you. Okay. Flowcharts. All right. There's a whole chapter in the back of your book with flowcharts. And to be honest with you, I wouldn't recommend spending a ton of time on the flowcharts. But there's 2 times that flowcharts are going to be super important. Number 1, with the gram-positive cocci. And the reason is because of all the hemolysis. Okay? So we've got alpha hemolytics, beta hemolytics, gamma, catalase positive, negative. And those little clues are going to be in the questions. So with the gram-positive cocci, that's going to be really important. And who else am I going to recommend a flowchart for? The Enterobacteriaceae, right? The Enterobacteriaceae. Lactose positive, lactose negative, H2S positive, modal, H2S negative, non-modal, all that good stuff. Okay? Yeah, so let me, Maria's asking about pages, so let me just briefly look back here. Yeah, yep, 500, okay? Now, the micro book, we'll take a couple minutes because we may as well do this and then take a break. But this is kind of important. So I'm going to teach you through chapter 6, which is the parasites, right? So I told you we're going to have a nasty good time with parasites on our last night together. That's going to be fun. But then the micro book keeps going. So this is really cool. So we get through parasites and we get to chapter 7. Chapter 7 is all of the systems. So here, this is a really good chapter to kind of quiz yourself. And it's got all kinds of little vignette clues in there for you. So as you're looking at that chapter 7, you can see based on body system, type of infection, what are the key clues are going to be in that table in the middle. And then all the organisms are listed. I can't figure out how this camera works. All the organisms are going to be listed on the end. So I would cover up the organisms, right? And then start to try to quiz myself as to what causes what. So here's where we take all of the bug parades and everything else we talk about, and we have it now nicely organized in the systems. Chapter 8 is called Comparative Microbiology. And, you know, that one can also be something you quiz yourself with. And here's why. All right, I'm going to make another good case for you. So in this chapter, we take all of the common features that bacteria have and we lump them together. So anything that's acid fast will have a list. And if you think about those clues, let's talk about this. You already know chapter 2 is a monster. And then it's not the only chapter of organisms, right? We got a ton of viruses, fungi, and parasites we have to go through. If they tell you in a vignette that something's acid fast, that takes all of that information and narrows it down. And now in your little mental Rolodex, you're like, oh, it's acid fast, so it has to be one of these things, right? It has to be TB, it has to be leprosy, cryptosporidium, right? But we're just going through that mental Rolodex in our brain. It makes it a ton easier, doesn't it? Absolutely. Okay. So that's important. Then you get the flow charts and you know, so the back chapters are really just good for you as far as studying, right? And testing yourself. So that's the kind of stuff you can really quiz yourself on. All right. So we might as well go ahead and take a break here, and you guys can think, you can digest some of that, and then we will come back and we will talk about staph and strep, right? That sounds good to me. The cocci, Steve, and the gram-negative rods, the enteric organisms, are the two that you really need to know. All right, break time, or as Abdullah would say, pizza time. All right, guys. So I was kind of stalking your chat a little while I was sitting here drinking my tea. And you guys are absolutely right. So doing this together is going to make it a whole lot easier. And you're absolutely right. Medicine is all about teamwork. And now we have this great stuff like Skype and FaceTime. And even Facebook, you know, just to like interact, even just through chatting, stuff like that. Really good ways to do this and kind of just quiz each other and go back and forth. It'll make it go a lot smoother. Absolutely. All right. So we know the gram-positive cocci. We're going to talk about staph and strep. We know that catalase is going to be an important way to differentiate between them. But before we get into talking about them specifically, what we're going to do is just kind of take a look at some of those lab features. All right. But again, here's where the flow chart's going to kind of be important. All right. Now, let's talk staph. So there's three big species here that we're going to talk about: Staph aureus, Staph epidermidis, and I love when people mispronounce that, Staph epididymis. I just kind of laugh. And then there's Staph saprophyticus, right? So the three major species, of which, of course, Staph aureus is going to be the most important. I know, it makes me laugh every time. I had a student just yesterday in class raise their hand and answer Staph epididymis, and it was all I could do not to crack up. Okay. So Staph aureus is going to be the most high yield, but regardless, all three species are catalase positive, but Staph aureus is coagulase positive. It's the only one that's coagulase positive, and it's the only one that's hemolytic. It's beta hemolytic. Now, when you start thinking again in your brain about organisms that are hemolytic, you think, you know, group A strep, you think strep pneumo, you know, all different hemolytic patterns, and sometimes you forget that Staph aureus is beta hemolytic, but here's why it's important, okay? Because again, sometimes those little clues in the vignettes can be a little misleading. So what if I tell you that I have, you know, some disease, and I say gram-positive cocci that were beta hemolytic were isolated? Now, your brain goes to group A strep. My brain kind of does too, but we can't forget about Staph aureus because that's a correct description, albeit a not complete one, but it's a correct description of Staph aureus as well. Also group B strep if we're really being inclusive. So you want to remember that Staph aureus is beta hemolytic. Also, Staph aureus is the only one that will ferment mannitol. Now, how we look for that is on something called mannitol salt agar, and if it ferments mannitol, it changes the color of the colonies from like a white color to a yellow color. And, you know, that's how a lot of those fermentation media work. There's a pH indicator, so when the fermentation happens, the pH causes the color change, right? Okay. And lots of different diseases caused by Staph aureus that we will plunge into very quickly. The other two species are both gamma hemolytic, which is kind of a pain in the butt, right? Gamma hemolysis is a little bit of a misnomer because they're non-hemolytic. They're not hemolytic at all. So gamma hemolysis is no hemolysis. Yeah. They're both catalase positive, coagulase negative. When we think about Staph epi, we think, I mean, at least in my brain, I'm thinking sometimes endocarditis, and I'm thinking prosthetic devices, and I'm thinking catheter-associated UTIs. Now, Staph saprophyticus, where that's going to be important, is when we start talking about urinary tract infections. It's pretty common, but overall, who's most common in a urinary tract infection? Right, honeymoon cystitis, right, is females, absolutely, and E. coli. So you hit all the high points, right? Now, Now here's going to be something important, because I know we like these buzzwords and such, but if you get a vignette about somebody with burning urination and frequency and a little hematuria, and they say that they just returned from their honeymoon, and that's all they tell you, what answer are you going to put down? Just ease my conscience a little. I was like, nothing. I'm not putting anything down. No. E. coli. E. coli is always most common. So the honeymoon, don't let the honeymoon kind of distract you, okay? Because E. coli is always most common. Now, where you're going to see saprophyticus is usually in sexually active young females, right? Absolutely. Abby's got it. Unfortunately for society, this no longer happens on the wedding night, usually. Okay? Now, that's why you really don't want to let this distract you from what you're answering, right? Because we just need to know over 85% of the time it's E. coli. Okay? Now, staph saprophyticus, yes, in young females usually. And when are you going to answer staph? Only if they tell you they isolated gram-positive cocci, right? Otherwise, you're still picking E. coli. Now, how would you differentiate between staph epi and staph saprophyticus, being that they're both catalase positive, coagulase negative, and gamma hemolytic? You have to use antibiotic sensitivity and resistance. So novabicin is the antibiotic that we would use for that. And staph epi is sensitive to the antibiotic, and staph saprophyticus, right, is resistant. Now, clinically, again, they can both cause urinary tract infection. So my guess is if they ever, and again, we know they're mean, but if they ever gave you a question where they brought you down to having to pick between the novabicin, it's going to be in a UTI. But in general, okay, in general, staph epi is catheter UTIs, and saprophyticus is just community-acquired kind of UTIs, right? But again, they could make you differentiate between them based on a urinary tract infection case. Absolutely. Okay. Now, as we start to get into the streps, again, for both staph and strep, you're going to think about the flow chart and some of these lab tests. But strep and enterococcus are all catalase negative. They are all coagulase negative. Now, let me just bring you to probably a helpful point that'll maybe help you. When you're faced with a vignette or a question differentiating staph and strep, look at the case, right? If the organism spread really fast and it's causing, a cellulitis or necrotizing fasciitis, something like that, it typically is going to be strep. And that's because strep has a lot of virulence factors that allow it to spread through the tissue. Now, because Staph aureus has coagulase, the staph infections are generally more contained. So we see boils, furuncles, carbuncles with staph because of the coagulase. All right. Hemolytic patterns are going to be important here. So we know that already. And we already mentioned who was beta hemolytic. So we know it's group A strep, group B strep, and we're not forgetting Staph aureus is also beta hemolytic. All right. Now, how would we differentiate between group A and group B strep, and really would we ever have to? Mostly they cause different types of infections. But in a lab, if you're really trying to get down to the causative agent, you could differentiate them again based on antibiotic sensitivity, where group A strep is sensitive to bacitracin and group B strep is resistant to bacitracin. But clinically, they cause different things, right? So, unless something goes really wrong and we have some type of bloodborne infection, or, group A strep, we're thinking strep throat, scarlet fever, rheumatic fever, glomerulonephritis, impetigo, but nothing that looks like neonatal sepsis and meningitis. Okay. So those in a vignette form, we should we should be able to differentiate based on symptoms, right, and what the disease is. All right. In the next group, strep pneumo, which I can't stress enough how high yield it is, clearly by my dollar signs right there in the table, is alpha hemolytic. Now, you guys tell me, because sometimes they put this in a question, what does that even mean? What is alpha hemolysis? It's partial, absolutely. So it kind of sort of lyses the blood cells, but it's not a complete hemolysis like we see in beta hemolysis. Okay? So on a blood agar plate, partial hemolysis looks kind of green. And strep pneumo is the major bug you want to think about. When you see gram-positive cocci that are alpha hemolytic, like 99% of the time, I'll want your brains to go to strep pneumo, and they will. But the viridans strep are also alpha hemolytic. Why is that important? It's not even listed in the disease presentations, but I know somebody that this happened to. They actually got pneumonia from aspirating the organism and got pneumonia from the viridans strep. So here we have somebody that has pneumonia from viridans strep that's also alpha hemolytic. So here's where you have to be a little bit cautious, okay? So in reality and on USMLE, you're picking strep pneumo most of the time, but I really just want you to read the question, because it's the optochin sensitivity that's going to help you differentiate between those, right? Where strep pneumo is sensitive to optochin and the viridans strep is resistant, right? Reality is even more tricky. Step one, reality is not really reality, right? It's kind of pseudo-reality. And then your reality, when you're actually dealing with patients, that's kind of what we're all here for, right? So that's where that optochin test can really be important. Enterococcus, we said, is used to be considered a strep. The hemolytic pattern on there is not really going to help you because it can be alpha, beta, or gamma. A lot of the times they're alpha and gamma. I don't know too many strains that are beta hemolytic, but they can be, and that's the point. So they're not as helpful in differentiating between them. And the last one that I want to mention that's not on here, but it's a pretty important fact, and we'll eventually add it to the notes. But if I had somebody that had endocarditis with Streptococcus bovis, what else would I be concerned about for that patient? I would be concerned about colon cancer. So strep bovis is an inhabitant of the GI tract. When you have colon cancer, that strep gets dislodged, and you can get blood infections, you can get endocarditis. That's Streptococcus bovis. So I'll just put strep bovis. So strep bovis. So that one we're going to add to the notes when we revamp them, but I didn't want you to forget about that one because that one's majorly important, because that's probably what they're going to ask you. If they tell you that you have a patient with strep bovis endocarditis, they're going to ask you to check them. They're going to say, what would you do next? Well, you're going to check them for colon cancer, right? So that's pretty high yield stuff in reality, and on USMLE. So that'll be important as well. Okay, So let's dive in to some of the bacteria. So we're going to talk about Staph aureus predominantly. Everything about Staph epi and Saprophyticus we already talked about, so we're not going to dive in any deeper to that. But Staph aureus we know is pretty high yield, so we're going to focus mostly on that. Okay. So what are some distinguishing features about Staph aureus? We know it's beta hemolytic. We know they're catalase and coagulase positive. We know all that, but that the top bullet point there is actually pretty important, because when we're trying to look at bacteria, the ones that are pigmented, there really aren't that many of, right? We know Pseudomonas, that's a big help because it's green. And Staph aureus is actually yellow, so it'll make some yellow colonies on blood agar. There's another gram-negative enteric called Serratia that's red. So, but there really aren't very many pigmented bacteria, so that's kind of an important point. So let's just take a look at some of those. So here in the middle, you can kind of see the yellow colonies, and that's nice. That's a good little tidbit there. Aureus means gold. That's awesome. So that helps you remember the pigment. You can see all of that clearing around those colonies. That's showing that the organism has lysed those red cells. Over on the left, that's kind of a classic coagulase test, where you inoculate rabbit serum with the bacteria. And if it's Staph aureus and it has a coagulase, it causes it to clump or make a little clot. And that you could compare to a negative organism that's not going to cause that serum to coagulate. So colors, you want me to repeat the colors? Staph aureus is yellow. Serratia, let me just type it in the chat. Serratia is red, and Pseudomonas is green. Very good. Thanks, Tatiana. She got me. She got my back. All right. And then over here, what are we showing? We're showing catalase, right? So those are clearly nice yellow organisms. We added the hydrogen peroxide, bubbles everywhere. That's clearly the catalase test. All right. Normal flora, we know that. Nasal carriers most of the time. Can be on the skin. A lot of people will transiently carry it on the skin, if you know what I'm saying. So if you're a carrier, really, that's how we're going to transmit it, right? And so the primary means of prevention of this is going to be washing our hands. Absolutely. Okay. So transmission, for the most part, hands, and there are other ways, of course, of transmission. Staph aureus we'll see a lot in surgical wounds. Does tend to contaminate food. The classic vignettes, if they're talking about staph gastroenteritis per se, are usually what they call the picnicky foods. But to be honest, it can pretty much grow in just about anything. But those are all pretty rich foods. It makes a good media for the bacteria to grow and then make the toxin. Right, canned meats, yep, you got it, all that stuff. All right, predisposing factors, surgery, absolutely. We talked about the foreign bodies, didn't we? Supersize me. Severe neutropenia. But that is going to predispose quite a few patients to other things as well. You know, we've got fungi to worry about there and other bacteria. IV drug abuse, CGD. So those are all just some risk factors for infection. Okay, now pathogenesis. So here's the things we want to focus on for Staph aureus. Now, when we talk about virulence, and this is a perfect example, okay? I can't necessarily give you, if I just asked you, what's the most important virulence factor for Staph aureus, you should ask me a question back. And that is, well, what disease is it, right? Because just like real estate, these questions about primary virulence factors and most important virulence factors are all about location, location, location. Okay? So am I in the GI tract? Then it's my enterotoxin. Am I causing toxic shock? Then we know what that is. If I'm E. coli and I'm in the urinary tract, you better answer pili, right? But if I go down to Mexico and I drink the water and all of a sudden I get watery diarrhea like I have in the past, then it's probably going to be a toxin, right? Location, location, location. All right. So the questions about virulence are very dependent on what the actual infection is. All right. Now, so some of the virulence factors. Protein A, mentioned this already. And I just wanted to kind of give you a visual of what protein A does. It actually binds to the Fc portion, hopefully you remember what that is, of the antibody molecule. So now you can see staph, and you can see the antibodies bound to staph, but what is the problem with that? What? I mean, you got antibodies that's bound to staph, but what? It doesn't look right, does it? It's backwards, absolutely. It's backwards. It's this part that needs to be bound to staph, not the Fc portion. The Fc portion is what we need for opsonization. So you're absolutely right, guys. The protein A inhibits phagocytosis because it interferes with opsonization. So if they asked a question about what virulence factor interferes with phagocytosis or opsonization for staph, well, then that's going to be protein A. Okay, enterotoxins. These are, of course, going to be important when we're talking GI issues with Staph aureus. Very, very common type of what we call food poisoning, and we'll get into that in just a little bit. But these toxins are very, very quick, and they are stable in heat. So the classic scenario, and I don't want to dissuade you from going out to eat because we all do it, but is that you've got a food worker that carries staph and they're not really clean, and then they're playing in the food, blah blah blah. Now the staph, I know it's disgusting, but we got to talk about it. And then the bacteria start growing in the food, they start making toxin. Okay, now staph carrier restaurant worker makes your food. Okay, and the cooking process kills the bacteria, but not the toxin. The toxin's heat stable. So we are ingesting toxin, and that's why it's fast acting. We ingest the toxin directly, no bacteria, we get sick. It is the double bucket, Nishat's got it, so he must have had it before. I've had it before. It is not pretty. And correct me if I'm wrong, Nishat, but whatever bucket you decide to pick is always the wrong one, right? It's messy. It's absolutely messy. But anyways, when we talk in general about food poisoning where we're ingesting toxin directly, the incubation period's usually short, right? Maria's like, how about on a plane? Yeah, I've been there too. We'll talk about it. But so the incubation period for this, the average time is about 2 hours, but I've had this and been sick within 30 minutes, right? So so that's what we're going to think about. We're going to talk a lot about diarrhea, which I know you're excited about. I'm just excited I didn't get it on the cruise that we went on. But if the incubation period is relatively fast, then we're going to think Staph aureus. We're going to think Bacillus cereus if we had fried rice. We're going to think something, that's an intoxication. Okay, toxic shock syndrome toxin, I know you know about because we killed it in immunology. We know it overactivates the immune system. We've already talked about that. So those are three major virulence factors. Coagulase, again, kind of keeps the infection relatively contained, like we said, furuncles, carbuncles, boils, but not spreading into necrotizing fasciitis. The cytolytic toxin, again, punches holes, but not really super important for staph. And the only time you're ever going to pick, obviously, the exfoliatin is if you got a baby with scalded skin syndrome, right? So that one is also pretty low yield. All right, so let's take a look at diseases. We talked a little bit about gastroenteritis. You can see it's within 2 to 6 hours, but on average, it's very, very quick. So around 2 hours, because again, we're just ingesting the toxin. We do have vomiting and diarrhea, hence the 2 buckets or double bucket. And again, I'm just in general not a lucky person. So if I'm going for this bucket, it's not the one I needed. But anyways, it's very, very quick. Okay. Now, what would we do to treat this? We wouldn't give antibiotics because there's no actual infection here. Okay. There's no infection. And so we're just rehydration, right? Okay. We're going to talk about a lot of organisms that cause endocarditis. Some are acute, like Staph aureus. So we have acute symptoms. They happen pretty quickly. Staph aureus, we're going to think about for IV drug users. And it is a pretty pathogenic, virulent type of endocarditis. So if you got, a young person, whatever, they're an IV drug user, and this endocarditis can take them down pretty fast. Okay, so this one is definitely acute, as opposed to a lot of other ones that are subacute, which are hard to diagnose. The symptoms aren't really overt, and we really have to kind of do a lot more to diagnose those. Abscesses, mastitis, all those things, again, we know that staph, the coagulase, is going to be important there. All right, toxic shock syndrome. We've mentioned numerous times in immuno, a couple times already tonight, but the symptoms of toxic shock, I can't even mention a transcontinental flight. No. But again, we have the sunburn-like or scarlatiniform rash. So looks like we have a sunburn. We've got on the palms and soles, and you can see a picture here, kind of peeling. So we got the desquamation, okay, and massive drop in blood pressure, multi-organ failure, and death if untreated. All right, toxic shock syndrome toxin is the culprit. And because the disease itself is toxin mediated, we can't always treat people with this, right? Because we give them antibiotics, we release more toxin. Sometimes the patient gets sicker. All right, so this one's pretty serious. Of the ones listed here, okay, toxic shock syndrome, obviously the most high yield. I would probably say followed by gastroenteritis, okay? Not gastroenteritis, I'm sorry, endocarditis. So of the ones that are here, toxic shock is going to be number 1. Probably acute endocarditis, I would say number 2. Gastroenteritis, there's a ton of other ones they may ask you. It sometimes shows up, but abscesses, mastitis, things like that, yeah, probably a little bit lower yield. All right, what else? Geez, that wasn't supposed to. Okay, we're gonna have to make that go away. But impetigo used to be pretty much considered strep, but now it's just about as equally staph and strep. And they're classic kind of lesions that you see there, and that's where they're going to ask you, what's the easiest way to differentiate between these two? It's the catalase test. Okay. Can we get rid of that? All right. Now pneumonia, we don't like think about Staph aureus a lot of the time causing pneumonia, but it absolutely does. And where you have to be really cautious of it is in nosocomial situations, right? You have a hospitalized patient, then staph is all over the place. And so we're really going to worry about it in hospitalized patients. And it is a very virulent pneumonia. So it's got a pretty high mortality rate. When I think about staphylococcal pneumonia, I will think about this. So let's say you had an elderly patient that got the flu, and they were very dehydrated, and you had to hospitalize them. So now they're in the hospital, right, getting better, but now they get staphylococcal pneumonia, a secondary bacterial infection. So as Miguel just said, thinks it's the most common cause of pneumonia post-viral infections. That and probably strep pneumo, I would probably agree. Okay. So those two are the ones you definitely want to think about. Nishat says salmon-colored sputum. Absolutely. When we think about sputum, we get to talk colors, which, ladies, we like colors, right? But not necessarily colored sputum. So we know with strep pneumo we have rust-colored sputum. And then what kind of sputum do we have if we have Klebsiella pneumonia? It's kind of orange. All right, so currant jelly. So it's red like a currant berry, but it's thick and nasty. So salmon colored is kind of orange. So guys, you would probably say orange. Girls, we like to list all of the different shades of colors, right? So like we know the difference between like pink and fuchsia and all of that, and guys are just like, oh, it's pink, right? So yeah, and Pseudomonas, green sputum, absolutely. So sputum color is gonna be a little bit helpful to you. All right, surgical infections. Doubt they're gonna ask you about surgical infections. Benjamin, that's awesome. It's the newest color from Revlon. That's awesome. Klebsiella currant jelly sputum. All right, surgical infections. It's common, but you may have that. I'm guessing that's going to be way more important on step 2 and 3 than on step 1. Osteomyelitis, yeah, you could see an osteomyelitis question from Staph aureus because it's the most common cause. And mostly we see it in kids, and you can see it in adults as well. But osteomyelitis in general, we think staph. If I'm a sexually active adult, it may change a little bit, right? You can start thinking about gonorrhea, maybe, potentially. But think about the age of the patient, kind of what they're doing. But again, that's going to be common. Salmonella non-typhi is going to be what? Sickle cell. Yeah, sickle cell osteomyelitis. Okay. Now treatment. So there's 2 basic things we need to know about treatment. Okay? Number 1 is if they tell us, or if they don't say anything about it being resistant, treat staph with NAF, right? Nafcillin, which is a methicillin antibiotic, generally considered the treatment for Staph aureus. On level 1 for treatment for staph, if they tell you, usually they don't come straight out and tell you. Maybe they'll show you a little gel with a little, somebody carrying the MRSA gene or something like that. However they tell you, if it's MRSA, you're treating it with vancomycin. Okay? So on step 1, that's going to be how we tackle staph. NAF for staph, and then vancomycin for MRSA. Not on step 1 are they going to ask you about vancomycin-resistant Staph aureus, okay? Maybe vanc-resistant Enterococcus, because that tends to be very resistant to vancomycin. But right now, Staph aureus isn't, and this is a good thing, the majority of strains are not vancomycin resistant. We do have drugs that can treat them, and I put a little flow chart, I think, together for you. And so you can kind of write this down. This is more for, I told you for what you're going to do for step 1, okay? And then this is more for your knowledge post step 1. But if we're talking Staph aureus infection, right, your streptogrammins, yeah, absolutely. So what this means, so just a little flow chart. So is it methicillin sensitive or resistant? And that's as far as they're really going to take you. If it's sensitive, you're going with the methicillin drug, nafcillin, oxacillin. If it's methicillin resistant, this part of the flow chart you're not necessarily going to worry about. You're going to go straight to vancomycin. Okay? So that's for step 1. That's how you're going to answer it. Sensitive to methicillin, you're going to use methicillin. Resistant, you're using vancomycin. All right. If it's hospital acquired, in reality, you're going to vanc. If it's community acquired, you can start with a lower level drug. So CA stands for community acquired and HA, hospital acquired. And then if they are severe infections, if they're resistant infections, then we're going to go with combination therapies, like you guys were saying, linezolid, the quinupristin, dalfapristin. But at this level, they're not going to take you on step 1. Okay? So that may be something they get into. I'm guessing, because I know this is going to sound really geeky and I probably shouldn't even tell you, but when Dr. Seastone was studying for his IM boards, we would do the questions together from the question banks. I know, that's how we would have fun for an evening. It's embarrassing. Anyways, but that's kind of the level that you'd see on IM boards, right? And all of this would be really then important, right? Is it community acquired, hospital acquired? Because those questions, boy, those questions are a lot harder. All the answers look really good for those. So I'm just building you up, right? Giving you the information you need so you can sit down with your loved ones and go through QBank questions at night for fun. I know, we're all nerds at heart. Okay. So that's pretty much it for staph. Miguel's not a nerd. Right, Miguel. Anyways, so now we're going to talk a little bit about strep. You can see in the picture, you can see strep, little chains there, but again, we addressed this already. You can never, it's never enough to differentiate staph from strep just based on that Gram reaction. So that's why catalase and hemolysis and all that is going to be more important. As far as the genus goes, we know that they're all catalase negative, and we group them based on these different carbohydrates in the cell wall. So we have group A, group B, group D, and we can additionally serotype them based on other properties they have. So for example, strep pneumo can be serotyped based on the capsule. So there's many different capsular serotypes. And where that is important, sure, Namrita, I can do that for you. Somebody going to have to email and remind me, though, because I'm probably going to pass out after this class tonight. So just send me an email. I'll put my email up at the end again. Okay. So strep pneumo, again, can be serotyped by capsule. Where that's important is when we're talking about strains that are more invasive and more pathogenic. because those capsular serotypes are going to be the ones that are actually included in the vaccine strains. Strep pyogenes, it can be serotyped by the M protein. Somebody said they had a question about M protein, right? And we know that that's anti-phagocytic, but certain serotypes of M protein are also associated. Oh yeah, you can do that. That would work too. As long as you get it written down. However it gets up there, it gets up there. But certain serotypes of the M protein have a higher likelihood of causing, let's say, glomerulonephritis, for example. All right. So that's just some genus features. We're going to talk about all of these different organisms because they're all, they're not all about the same as far as high yield, but questions can show up on any one of these. So we want to make sure we hit all of them. So we'll start with group A strep. Group A strep, we know strep throat, we know all that. Yeah, Moses, you're absolutely right. The most high yield is strep pneumo by far, absolutely. And we'll kind of go through why that is, but it is pretty important. Okay, we know it's beta hemolytic. We know it's sensitive to bacitracin. There is an enzyme called pyrrolinidyl arylimidase, PYR, that's positive in group A strep and enterococcus. I don't think that they use that. I haven't had any students come up to me and said, oh yeah, they definitely put that on the test. I mean, it's a newer thing that UWorld does, of course. UWorld's going to bust your chops and make the questions as hard as they possibly can, so you think you don't know anything and just go back and study it all again, right? But PYR, you're right, Miguel, is just an additional clue. So group A strep and enterococcus, yeah, they both have it, PYR. So just an enzyme, no significance that you need to worry about for that. Reservoir for strep, we know in general the throat can be a transient on the skin. Oh, that's good, George. So you guys come up with the best kind of mnemonics there. How do we transmit this? Mostly by respiratory droplets, but it can also be direct contact. All right. Now here's where you start to panic a little. When you see a list that's a page long of just virulence factors, I know you want to shut the book and go away and eat pizza, right? But don't worry about it, because I'm going to tell you the ones you need to know, number one. And number two, you just want to kind of know in general how some of these things work and why they're important, but you certainly don't need to know every one of them. For example, the hyaluronic acid, that's the capsule, but it's non-immunogenic. So as far as what it actually is doing for the bacteria, you really don't need to know. All right, so we're not going to worry about that. All right, M protein for group A strep, probably, unless we're talking scarlet fever, I would consider it probably the major virulence factor. And again, it disrupts complement activation, anti-phagocytic. Again, the M12 serotypes associated with glomerulonephritis. It's one of the more major virulence factors for group A strep. You've got a couple different homolysins. One's immunogenic, one isn't. Doesn't really matter. You're just going to use those to look for a rheumatic fever. So if people have positive titers of the streptolysin, but other than that, you don't need to know anything really about the streptolysins. All right, now these are all what we call spreading factors. So think about group A strep and think about what it does in the skin, right? It spreads. I mean, necrotizing fasciitis, I can't think of anything more horrible. And of course, group A strep's not the only one that does it, but I use that as kind of the guide because this bacteria makes stuff that allows it to spread really fast through the tissue. It liquefies pus. It breaks down fibrin. I mean, it essentially just, boom, it just like totally aggressive moving through the various tissues. So they're not going to ask you about any one of those specifically, but just knowing that strep can do that helps you kind of keep your eye on certain diseases. No mnemonics there unless George has one. George, help me out. Because again, they're probably not going to ask you anything specific about those. Okay? All right. Now, here's another one. So I put it in red. Exotoxins. That's going to be important. Where is it going to be important? In scarlet fever. Okay? You give them a fever. But scarlet fever, it's going to be important because it's actually the toxin giving you the symptoms. Okay? So I got a kid with scarlet fever. We'll talk about symptoms in just a minute. And I ask you about a virulence factor. You better pick the exotoxin. All right. I love the jaws. You know I do. So keep them coming. All right. And potentially a super antigen. But again, we're going with toxic shock. Correct. Can this one cause toxic shock? I don't know that it ever gets that far, but it functions in the same way. So potentially, I guess, but I've never really heard of it doing that. But I guess because it has the same mechanism, potentially it could do that. And we know USMLE writers are jerks, so just keep that in mind. But clearly they're probably going to ask you about staph. Okay? All right. The other thing I'm just going to point out to you, because it's the annoying stuff we have to get to with the genetics, is that there are quite a few virulence factors that bacteria have picked up because, and this is going to sound really weird, right? Because they got infected with a virus that is specific for bacteria. Okay? So it's like giving them superpowers. Like it's like Spider-Man, right? He gets bit by a spider. He's now superhero Spider-Man. So these bacteria get infected with a phage, and now they're like superheroes as far as bacteria are concerned because they're more virulent. Now, Teal, you're exactly right. That's transduction. But as we go through the organisms, you're going to want to kind of keep your eye on when it says phage encoded. Those are the ones they're going to ask you about. So here's a typical question. All right, here's a typical question. I give you any simple question about strep pyogenes, scarlet fever, sandpaper rash, strawberry tongue, nausea, vomiting, all that good stuff. And I say, how did the bacteria in the above case acquire its virulence? And you're like, son of But then you remember, Muscatello. She told us about this. She told us to look out for these. That's the exotoxin. We got it by a phage, which was by transduction, as Teal said earlier. And we're going to go through all that when we get to the genetics chapter. All right. Let's just get through some of these and then we'll take a break. Pharyngitis, right? Strep throat. Too easy. Not going to ask you a question about strep throat. What they're going to do is give you a question about glomerulonephritis and tell you that the patient had a sore throat, but they're not going to give you a straightforward question on strep throat, right? It's too easy. And besides that, as I can attest to right now, because I'm pretty sure I have one, viruses are more common in general as cause of pharyngitis. All right. Scarlet fever, yeah, they can absolutely ask you about scarlet fever. That's when the toxin's going to come into play, and either how the bacteria acquired the toxin, but that one, and pretty much the rest of these are all, people do get scarlet fever. My kid actually had scarlet fever. My son did, because, we call Brian, I told you this, the honey badger. And he, so he doesn't really complain. Clearly he had a strep infection, but we didn't know he had a sore throat. And then all of a sudden, I went to hug him, and that nice soft baby skin that you're used to hugging, wasn't there, and his skin felt like sandpaper, all of it. So it was horrible. But then I knew he had scarlet fever, and so people get scarlet fever all the time still. And what's the only reason we treat scarlet fever? Because scarlet fever ain't going to kill you. But why do we treat patients with scarlet fever? Well, that's right. To prevent glomerulonephritis, rheumatic fever, all that, right. We don't want it to get any worse than just scarlet fever. So you're absolutely right. All right. And you can see the symptoms there. I'm going to show you some pictures. We know impetigo. We know all the spreading types of skin lesions. That's pretty low yield. What's really high yield are these. And we talked about these, of course, in immunology. Rheumatic fever, which you got to know is a type 2. Glomerulonephritis, which we got to know is a type 3. Okay? So we went through those. And they can ask these questions again and get at them from multiple ways. So they can give you the symptoms of rheumatic fever, ask you something about the bacteria, ask you something immunologic like the hypersensitivities, ask you about the mechanism, what the mechanism of the type 2 is versus the type 3. So they're going to come at these two diseases from all different kinds of angles, and you're ready for them, right? You got the eye of the tiger. So we're not going to let those get to us. So, but those are really high yield. And again, they're not going to ask you a question about strep throat, but they're going to give you one of these and say your patient had a sore throat. Jerks. Jerks. Okay, so just kind of go over strep throat. Obviously, got a lot of inflammation, nice little pus pockets back here. Yeah, one of those pyogenic infections. All right, this is scarlet fever, and you got the strawberry tongue. Okay, so tongue kind of looks like a strawberry. You do see some erythema in the groin area with this. You can't really see the sandpaper rash. You can feel it though, and it feels like they basically have goosebumps everywhere, right? But it really does feel like sandpaper. So those are going to be the things that should jump out as far as scarlet fever. Now, diagnosis. We know we have a rapid strep test. It's based on ELISA. Really a simple test, except for it can miss some infections. So we're going to go ahead and culture all of the negatives. Okay. Does the Kawasaki tongue? I'm not familiar with Kawasaki tongue, so you're going to have to, maybe you guys can chat about that, because we'll take a break in a minute. But so we're going to culture all negatives. That's important, because if we see something beta hemolytic, that's probably going to be, the group A strep. Here's where I said streptolysin O matters. You don't really need to know what it is, but we can look for titers of this to give us an indication of whether or not somebody has rheumatic fever. And then treatment. Oddly enough, the group A strep, so strep pyogenes, is really still pretty okay with the beta-lactam. So there's really not resistance so much built up for group A strep. All right. So the penicillins are going to work just fine. The beta-lactams, we can use macrolides if we have to, but your drugs of choice for these are still going to be your penicillins. All right. Prophylaxis, really, we have to consider prophylaxis in patients that have rheumatic fever, because obviously if we get this infection again, our immune system does what the second time around? It's faster, and it's going to exacerbate that disease. So we have to prophylax for that. Natalia's like, I can't. Yeah, I just wanted to get through Group A strep. We're absolutely going to take a break now. All right, guys. We went a few minutes over our normal break, but it was a good stopping point because we will leave the Group A streps and we will come back to the Group B streps. Okay? All right. So I will see you in 10 minutes. Alrighty. Just a little bit left, guys, so I'm jealous that you're at Starbucks. And I'm not, but we'll deal with that later. All right. I think we'll definitely get through the rest of these gram-positive cocci tonight, so let's move on. Group B strep, pretty much what they're going to ask you about Group B strep is what? Is neonatal meningitis. It's the leading cause still, okay? Even though we test for it, still considered the leading cause of neonatal meningitis, followed by E. coli. It's beta hemolytic. We know resistant to bacitracin. That's how we can differentiate it from Group A strep. Okay, and the thing that we often associate with Strep A galactiae or Group B strep is this crazy camp test, right? It's kind of a crazy test. It is not cyclic AMP. It's just kind of a weird random fact that if we culture Group B strep along with another organism like Staph aureus, an organism beta hemolytic, you get an enhanced beta hemolysis that you can see up here in this picture, and it kind of looks like an arrowhead. Yeah, it's kind of weird, right? Who discovers this stuff? You just kind of feel bad for them, right? Because this was like the most exciting thing they ever did in their life, right here. I was the founder of the camp test. So, but that's it, right? Camp test, that's all it is. Not cyclic AMP, but just enhanced hemolysis. Now, we know what the reservoir is for Group B strep, vaginal mucosa, but not in everyone. It's only in, you can see the stats there, about 15 to 20% of women can be also an inhabitant of the GI tract. That's probably where it got a little misguided, a little lost. Makes its way to the vaginal mucosa. Now, the reason that there's $4 signs, again, because it's the leading cause of neonatal meningitis. So that's one we definitely have to know. They're not, transmission. That one's pretty straightforward. Pathogenesis, they're not going to ask you. Diseases, neonatal meningitis and sepsis, most common causative agent. That's going to be your question. All right. As far as Group B strep is concerned. Now, as far as treatment, in general, when I think about Group B strep and really any bacteria that causes meningitis, we think about the third generation cephalosporins for treatment. Here, you can use ampicillin and an aminoglycoside together, or just the cephalosporin. Okay. So Natalia, so you bring up a good point. In neonatal meningitis, they're acquiring it from the vaginal mucosa during delivery. Okay, so I don't even, I guess it would be the very end of the third. But you can have placental infection, so this can cross the placenta, but it's not one that we ever really talk about. So we're going to think about it more for neonatal meningitis. Okay, so that's exactly through the birth canal. Okay. Prevention. So what do we do to prevent it? Well, we screen for this, and we don't. So let's say I was screened for Group B strep, and I was positive. They don't treat me right there. You're right. Listeria does too. Very good. Absolutely. But they're going to treat me during delivery. And they're going to treat me during delivery because if this is part of my normal flora, there's a very good chance it could come back. So we're just going to treat during delivery. No, it won't get transmitted to the neonate. All right. So that's Group B strep. Now, strep pneumo. I think you guys already said. I'm gonna back you up on this. Of all of the streps, this is going to be really the most high yield. And we also said that when you're thinking gram-positive cocci that are alpha hemolytic, you really want to think about strep pneumo. That's going to be the thing that should just boop, 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 like light bulbs should go off and sparks should fly. Except for you want to just remember about the viridans strep also being alpha hemolytic. So these guys are diplococci typically, so we'll find them oftentimes in pairs. We find them in the upper respiratory tract. Now, when we get down here to transmission, this is going to be kind of an important point for lots of different organisms that we're going to talk about over the next few days. It colonizes without causing disease a lot of the time, but only in certain individuals does it cause disease. Now, who is it going to cause disease in? It has lots to do with various host factors, immune status, lots of different factors come into play. But, people are carriers of strep pneumo and they may never get an infection by Streptococcus pneumoniae, whereas other people can get exposed and they can get ill. We see the same thing like with Neisseria meningitidis, Klebsiella pneumoniae, not considered very highly communicable, and people can live with them, they're just fine, and then other people we know tend to get sick. Now, that's interesting because strep pneumo is whatever disease pretty much we're going to talk about strep pneumo, it's the most common causative agent. Like, it's just the best at what it does. And most of that gets back to the capsule. All right, so it's got a good virulence factor, can avoid the immune system. Predisposing factors, kind of lots, but really anybody can get strep pneumo, okay, if all of the host factors are right. We mentioned staph causing a post-viral pneumonia, but strep we also said, so staph and strep are going to be probably your two most common, and you can see some of the other risk factors there. The only other, well, I guess I'll point out too, in an alcoholic, okay, so I want you to think, so if I give you a question about an alcoholic with bacterial pneumonia, lobar pneumonia, and I don't tell you anything else about it, I just say it's lobar pneumonia, it's bacterial, well, now you got to pick between strep and Klebsiella, all right? Strep is still more common. Strep pneumo is still more common. Now, what that does is it puts Klebsiella on the radar screen, okay? So now, whereas if your patient's not an alcoholic, Klebsiella is not even on your differential, but now all of a sudden they're an alcoholic and it is on your differential, okay? But strep's still more common. Again, USMLE test, they're jerks when they're writing the questions, but I would say at least 85% of the time, if they're writing a question about an alcoholic with lobar pneumonia, even those guys are going with Klebsiella, right? Confabulations. All right, then the other important point that we need to make, and I think we made it immunologically as well, but good to just review some of that. If you have an asplenic person, all right, so my students tell me all the time they get pimped on their rotations constantly, their surgical rotations about this. Surgeon's about to take out a spleen, and they ask the student, hey, what should we recommend as far as vaccines for this patient? Well, you're going to vaccinate them against the encapsulated organism, so strep pneumo, Haemophilus, maybe even Neisseria meningitidis, right? But those are the things that we're going to need protection against because they're more susceptible to those. Okay, here's another picture of strep pneumo. Again, sputum gram stain, a little bit different. You see all the neutrophils in there, lots of inflammation. Okay, but the capsule is going to be the major virulence factor. How does it work? It's antiphagocytic, it's sugary, it's slippery. If this is strep pneumo and this is the neutrophil, it slips right away. All right, so that's going to be the major virulence factor. And I honestly doubt they're going to ask you about any other virulence factors because this organism did not become virulent until it acquired its capsule. So that's going to be the primary virulence factor. Got a lot of others. It's got an IgA protease. That's good because it's a mucosal pathogen. Got teichoic acids that can help with attachment. It's got a hemolysin that gives you the rust-colored sputum, right? But the capsule is the most important, so keep that in mind for strep pneumo. Now, diseases. 3 major diseases for strep pneumo. Obviously, community-acquired pneumonia or lobar pneumonia is strep pneumo, most common cause, okay? Shaking chills, high fever, lobar pattern on chest x-ray, rusty sputum or bloody sputum. Klebsiella also causes lobar pneumonia. Yeah, they both do, okay? But again, if given nothing else, and we have a patient that has these symptoms, we got the x-ray, and they just ask you what's most likely, we're going to think strep pneumo, okay? Cavitary, when I think cavitary pneumonia, I think a lot of time you think like Staph aureus can do that. Right, TB can do that. Klebsiella renzo does cause a similar color sputum, but it's much thicker and the cart comes up. All right. Yep, even nocardia probably. All right. Now meningitis, and let's just we're going to add something here because we want to make sure we know what these are going to be. So I'm going to add to adult meningitis, we'll say childhood meningitis. So strep pneumo is the most common cause of adult meningitis, but also meningitis in kids. Okay? And meningitis is a weird one because age is going to be important for lots of different things, especially when we get into viruses. But meningitis is very much age dependent. Okay? So in neonatal meningitis, group B strep, most common cause. In kids, it's strep pneumo, unless I tell you that the child's been not vaccinated. Then it's Haemophilus influenza, right? So in kids in the U.S., if they're vaccinated, strep pneumo. Miguel is right if they're unvaccinated, Haemophilus, and they still ask those questions all the time. What about between the ages of 18 to 22? What's the most common cause of meningitis? Yep, Neisseria meningitis. Absolutely. And then back in adults and elderly, it's strep pneumo. What about, this is a trick question, immune compromised patients? You're right, Sarat, lots of unvaccinated. All right, so George hit 1, listeria. Yep. So in chemo or cancer patients and transplant patients, it's listeria. What is it, though, in AIDS patients? So here's just a weird thing that happens with meningitis. We got an umbrella called immune compromised, and mostly that it covers the same organisms. But here's a little difference. So we've got somebody with a transplant or chemo, it's listeria. AIDS patients, it's cryptococcus. So just a little different. But the meningitis is going to be very age and then host dependent. So this is, again, one of the most common causes in certain age groups. Otitis media and sinusitis in kids. For neonates, it was group B strep. Otitis media and sinusitis in kids, most common cause. Now, some of you may say, what? We vaccinate, and we absolutely do. we vaccinate, but we vaccinate against strains that are more invasive. So there's lots of other serotypes that can still cause otitis media, but we're vaccinating these kids so that they don't get pneumonia, so that they don't get meningitis. Otitis media, yeah, so we don't vaccinate every serotype under the sun. Not sure what you wanted me to repeat exactly, but what I was saying about immune compromised is that in AIDS patients, for meningitis, the most common cause is cryptococcus. And in other immune compromised patients, it's different, and that's different in general, right? You don't usually see that. So in transplant patients and cancer patients, it's not cryptococcus, it's listeria. All right, so let's go through them again. Neonates is group B strep. Kids and adults is strep pneumo. All right, so beyond the neonatal period, strep pneumo. Okay, you're absolutely right, Jose, on the differential for otitis media. Absolutely. And in the age group of 18 to 22, we said Neisseria. And then again, we've got it broken down in the immune compromised. AIDS patients different than cancer and transplant patients. So that's all going to be helpful because that's the information they're going to put in the vignette. And that's what they're going to use to get you to pick the specific answer. So we'll review that stuff when we get to the other organisms, but it's a good, it's a good starting point for meningitis because we know that that's going to be high yield. Okay. As far as diagnosis for strep pneumo, again, we've got a little bit of a difference in reality versus what necessarily happens on step one. So how on step one are they going to tell you that you have strep pneumo? 9 times out of 10, they're going to show you a picture of it. Okay? 9 times out of 10, they're just going to show you a picture. We did a sputum gram stain. We gram stain the CSF. Here it is. And you see the low gram positive cocci. All right, so that's one way. Another way on step one that they're going to tell you what it is that they're going to tell you that in sometimes stupid ways that the bacteria that they found has a capsule. And one of the old school ways to differentiate these encapsulated organisms would be to add antibodies to the capsule, and then you get a swelling of the capsule, and that was called the quelling reaction. So they don't really do that anymore, the quelling reaction, but sometimes people writing these questions are like 150 years old, and they think, oh, I got the best idea for a question, and I'm going to put that quelling reaction in there. And nobody bothered to tell grandpa or grandma that we don't do it anymore, right? Because sometimes they get pissed off when you say, we don't do that anymore. But it still gets into the question. So if you see quelling, yes, I'm serious. If you see quelling positive, it's not specific for strep pneumo, but essentially it just means that the organism has a capsule. And they do sometimes, I'm not even joking, they'll still put that on there. So you're going to want to be able to identify a picture, and you're going to want to know that the quelling reaction means we have a capsule. All right, so those are the 2 ways they're going to tell you, outside of just the clinical symptoms. All right, so we're going to treat differently depending on the disease. Strep pneumo is becoming more resistant to antibiotics, so in reality, depending on where you work, at which hospital, they will have charts as to how resistant strep pneumo is in your area, and then you pick the drug. Yeah, I can go back to the picture. Hold on. Let's go back to the picture because we said it was important. So here's a good example of the picture. Okay, now this is in sputum, but you can see the gram-positive diplococci, and what did I say was surrounding that? The nice white halo. So you've got, that's the capsule. Okay? In the CSF, of course, if it's meningitis, you're also going to see neutrophils, and you'll see you'll see the gram-positive cocci as well. Okay. Again, so in reality, treatment depends on where you work. On boards, a little bit different. For pneumonia, generally a macrolide to cover pneumonia. Meningitis, again, you're thinking third generation cephalosporin. Otitis media is an interesting one because, again, it really depends on where you live and resistance, but they'll generally start you with the beta-lactam and just kind of move up. I mean, in reality, a lot of the times they're giving Z-Paks and stuff for otitis media. You're right, augmentin is better, but it's rough on the GI tract, that augmentin. So usually they start kind of small and work their way up. Okay. Collateral damage. Absolutely. So prevention is, of course, that we have a few different, we do have vaccines for this, all right? But we have lots of different serotypes. Okay. So, interesting, we have two different formulations of these vaccines, and I think you'll remember, because I was pretty adamant about this in immuno, that the vaccine stuff is really important for USMLE. Okay, they love them some vaccine questions. But here's a little, it's just kind of different. So the pediatric vaccine now has 13 of the most common serotypes that are more likely to be invasive. Okay? Because if you think, okay, well, it's 13 of the most common serotypes, why are they still getting otitis media from strep pneumo? Because, well, we're trying not to give them pneumonia and meningitis, right? It's 13 now. It used to be 7, Manel. It used to be 7, and they just reformulated it pretty recently up to 13. So if you have, maybe an older version, it's now 13. The adult vaccine, 23 of the most common capsular serotypes. But just look at the two different vaccines. They're completely different. One has 13 serotypes and is conjugated to protein, which makes it a T-cell dependent vaccine. Hopefully we remember this. Haemophilus influenza type B, we know that's super high yield. Adult version, is just capsular polysaccharide. And so we're getting a nice, good, high titer of IgM that's protective, for a good period of time, but then we're going to have to give it, after so many years. I think the current recommendation is like 4 to 5 years. All right. So one is a conjugate vaccine, the other is capsular polysaccharide. Now, if I'm a jerky question writer, I'm going to ask you something about, the adult versus the pediatric vaccine, getting at which one's conjugated to protein, which one isn't, and then ask you something about the immune response, right? If I really have on my jerk hat. I don't know that they've done that, but, if I'm writing questions and I have to be a jerk, well, why not go for broke? You know what I'm saying? All right. Viridans, strep are pretty low yield, so I don't want to spend a ton of time on these, but we know these are kind of our normal inhabitants of our oropharynx. We know they can cause cavities. We don't like that so much. And they're also alpha hemolytic. So I said just be a little cautious as you're reading these questions. The biggest likelihood of you seeing this kind of a question is on somebody that has a damaged heart valve getting subacute endocarditis. Namrita is the conjugate vaccine, so it's going to be the pediatric one. Okay? So endocarditis in somebody with a damaged heart valve. So we know it can cause cavities. They're, again, probably not going to ask you about that. Probably going to be infective endocarditis, again, subacute. So a little bit more difficult to diagnose, and the symptoms aren't going to be as, prominent as you're going to see clearly with acute endocarditis. Now, interesting, I'm going to share something with you. It's a little bit of a sad story, but it's just so microbiologically crazy that you won't even probably believe it. So I had an uncle, so my dad's brother, and I don't know, some of you are Italian, I know, so you know that these Italian men are very stubborn, okay? So he had a valve replaced, probably, I don't know, 3 or 4 years previous to what I'm telling you now, okay? His wife goes out of town, and he broke a tooth. Not even kidding. He super glued it back to his mouth. I'm also not even kidding. Didn't want to go to the dentist. Thought, I'm not paying the dentist all that money to put my tooth back together. And he, honest to God, super glued his tooth back in his head. True story. Now, if you're super gluing your tooth, you probably really have to jam it up in there, right? I'm guessing, to get it even. And so that clearly causes the dislodging of some bacteria. All right, fast forward several months. My uncle, all I hear is, your uncle's having this horrible back pain. He can hardly get up. And, he passed out once. His blood pressure got so low, right? Well, long story short, he was having symptoms of both endocarditis, sepsis, and osteomyelitis from that stupid tooth that he glued back in his head, and it eventually killed him. That infection killed him. I'm not even kidding. Insane. So Uncle Joey, looking down on me, he's saying, I can't believe you just told that story, but I did. Anyways, it definitely helps you make the connection between teeth and heart, right? And I mean, really, at the end of the day, please don't super glue your teeth back in your head. Like, sometimes you do have to go to the dentist, right? Absolutely. Thank you for your apologies. But my Uncle Joey is now hanging out with my dad, so it's all good. I'm sure they're playing cards up there and calling each other every name in the book, right? All right. So that will certainly, you'll never forget that story. Treatment, penicillin and aminoglycoside, and if you do have the damaged heart valve, prophylaxis prior to dental work. Hence, if you absolutely have to glue your tooth back in your mouth, at least get some antibiotics, right? I know, it is sad. Sad and unbelievable. All right, last of the gram-positive, cocci, did the glue hold? It did. The glue did hold, right? I know, the moment when you're expecting me to tell a funny story, but I don't. All right. Okay, Enterococcus, all in all, pretty low yield. Melanie, the vaccine is only polysaccharide, so you're only going to get an IgM response. It's only going to be a B-cell response because it's not conjugated to protein. All right, so Enterococcus, 2 species. And the species name should tell you where we're going to find these. All right? Faecalis faecium in the feces. Okay. The 2 major things we're going to think about with these guys are typically surgical type of infections and urinary tract infections that are nosocomial. All right? So these are, a lot of times, these are endogenous types of infections, and some type of surgical procedure causes them to get released from the gut and then hit the damaged heart valves or another site. Okay, so endocarditis, UTIs, almost always catheter related. Again, pretty low yield, so we don't want to kill it here, but the big thing we just have to think about if we are talking about enterococcus in one of these questions is that they're very resistant to even vancomycin. And Staph aureus, that's vanc resistant, actually got that gene from enterococcus. So it really is kind of the leader as far as vanc resistance. Oh yeah, that's good, Miguel. Elderly with a prostate issue and enterococcus, yep. Absolutely, that would be a good vignette for this as well. So again, all in all, pretty low yield. I don't want to spend too much time on that. All right, now that's good. We got through all the gram-positive cocci. We get to change it up a little bit, right? So we get to talk about some gram-positive rods. Now, you've got this nice little summary table for you. At the beginning kind of section before we get into the gram-positive rods. I like the table because it's very good at putting things in groups for you. All right. So who are the spore formers? Well, you got to know those: Bacillus and Clostridium. Who's aerobic? And then you can see by the minuses who's anaerobic. So 2 of our anaerobes are in this category. Who has toxins? Who is a facultative intracellular? That's going to be important. And then if we move down, who can cause infections in immune compromised patients, right? We know which ones those are. And then acid fast. So here's a good a good kind of clue here. Nocardia is partially acid fast, which means it stains part pink and part blue. Mycobacterium obviously is completely acid fast, but in an AIDS patient, those 2 diseases can look similar. So I can have tuberculosis or I can have a nocardia infection. I'm still coughing up bloody sputum, and I still have acid fast organisms. Nocardia is much lower yield. You just have to, that's one of the jerky things you have to be on the lookout for. They could ask you a treatment question. And the simplest way, they're going to either mention something about being partially acid fast, or they're going to tell you that it's branching. Remember, these are branching rods. All right? So actinomyces and nocardia. Now, you see, which we didn't necessarily mention, that Mycobacterium is in this list. And the reason I didn't bring it up when we were going through all of the different gram reactions is because if you get a question on whether it's tuberculosis or leprosy or, marinum or any other, basically mycobacteria, they're just going to tell you it's acid fast. They are not going to tell you that it's gram positive. So where I want you to really think about Mycobacterium is under your acid fast organisms, because pretty much never they're going to refer to it as a gram-positive rod, but that's the classification that it's in. Okay, so now we're going to move on through Bacillus. Okay, in general, okay, we got 2 organisms here. Now, Bacillus cereus, they used to ask all the time. And they've kind of backed off on it a little. So it shows up still occasionally. It's one that you just really want to get because we know it's easy. And let's be honest, we know what they're going to ask about Bacillus cereus, right? Intoxication, fried rice. And do you ever notice it's always medical students that go to the Chinese restaurant? They always use you guys in the vignettes. That's crazy. And then there's anthrax, okay? Anthrax, of course, in general was pretty low yield because how many naturally occurring cases of anthrax do we have in the U.S.? Before 9/11, there were like 7 in the previous like 10-year period or something. So it was very, 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 very just inconsequential as far as disease. Miguel's got it though. Biological wars. Sounds like it. Oh, it is a good game actually. One of my students recently told me about a game that I got on my phone, which I should have never got, called The Plague. Right? You got to get that one. And the job is you have to infect and kill the whole world. Right? It's right up our alley. Oh yeah. Download it. It's crazy. But anyways, this is going to be biological warfare, right? So how are we going to know that it's an anthrax question? They're going to put it in the vignette of biological warfare. When did we see this in 9/11? We saw it in postal workers. We saw it in government buildings. Yeah. So with animals, so that's a good point, Maria. It is associated with animal skins. So potentially, also not just the fur coat, but drum heads that are made from animal skins. Just imagine you're beating the drums, and now you're aerosolizing all that. Andrea, no, mine is 9/14, and it's just a sad time of the year here. But we'll just henceforth know 9/11 as Andrea's birthday, okay? So we're not so sad about it. All right. Bacillus. So a couple things we need to focus on for bacillus. Number one, what do you see in this picture? Well, you see purple rods, and what the heck are those little balloons in there? Yep. Absolutely. They're spores. They are spores. So when you see spores like that, first of all, they don't gram stain, so they're just going to show up as those nice little white bubbles. And that means that these bacteria, it's not so conducive for them to live. So the media's getting the food's getting a little light, the environment's getting a little, where it doesn't like it, and it's getting ready to sporulate, right? So that's what those spores look like. These are aerobes, so they only grow in air. And this one, USMLE, because again, they're jerks. Oh, sure, I'll put the pointer at it. They like to ask about things that are different. So can you see the green arrow pointing right at the round spore? Okay. And you can see them within all of the different chains, right? And you see the boxcar, absolutely. You got it. You guys got it. I love how you guys help me teach the class. You make my life so easy. But notice that the capsule here is not a polysaccharide, it's a polypeptide. That's different than all the other capsules that we talk about. So again, if I'm a jerk question writer and I'm writing about anthrax and capsules, I don't know, maybe something there in the fact that it's a polypeptide. We know, of course, it's a biological warfare agent, and just like I love the fur coat thing, that's awesome. But animal skins, absolutely, in the soil associated with animals, and then, of course, as a biological warfare agent. They absolutely do ask this on step 1, as far as a biological warfare agent. And the question, I'm trying to remember, when they asked this in the past, at least from what I gather, it was a postal worker. So they'll put those clues in there for some of these questions. So first, let's talk virulence. Okay, we've got the anthrax toxin, and while it has a bunch of different parts, the part they're going to ask you about is the edema factor, which is the cyclic AMP inducing part. Okay, so the protective antigen is the part that binds to the host cell. The edema factor is the cyclic AMP inducing part where we start to get the edema. That's very common with anthrax. And then, of course, there's a lethal factor that no one ever talks about because all it does is kill the cells. But whatever. USMLE doesn't so much care about that. Okay. We're going to focus on two different forms of anthrax. Cutaneous, where you come in contact with the spores on your skin. That's going to be typically what we think about with the postal workers or anybody that's opening the envelopes, although they can still inhale the spores. But direct contact in the skin, cutaneous anthrax, where, again, the spore turns into a bacterial cell. The bacterial cell makes toxin. Toxin makes people sick. And I think somebody put up here earlier, although I can't remember who did it, the black Escher. So this is very common presentation for cutaneous anthrax, this black Escher, and there's edema associated with it, a very erythematous border, so lots of red and lots of edema. So that's cutaneous anthrax, which is not nearly as problematic as pulmonary or inhalation anthrax. So this is more mild. But if you're really talking about biological warfare, what you're really going for is pulmonary anthrax. Why is this such a good agent of biological warfare when we have antibiotics that can treat it? Well, here's the reason. You have to treat this within the first 48 hours. That's right. Or your patient's probably going to die. Why are they going to die? Why do we ever talk about virulence factors? Right here, okay? You got a patient with anthrax. They're a government employee, or at least around here, they work at NBC, right? Lots of those letters went to NBC, a broadcasting company. And you inhale the spores, they turn into bacteria. Now they start making toxin. I'm surprised it wasn't Fox too. And now, once the bacteria really get going and start making lots of toxin, now let's say you give this a few days to keep going. You treat with antibiotics, and now all of those bacteria start to lyse. Where's the toxin before we secrete it? It's inside those cells, and it whoosh, comes flooding out, right? So we kill these people from the toxin. That's why this is a good biological warfare agent, because if you don't catch it early, you can't really catch it at all, right? Your patient is probably going to die. Now, somebody, Mincho said mediastinal widening. That's the big clue, isn't it? That's the big clue, because otherwise, well, let's see, pneumonia, cough, fever, malaise, hello, like everything else, okay? How do we know this is anthrax? Because we get edema, and it shows up on that chest x-ray. That's huge. That's the big clue, okay? That, aside from the fact that, we're a government employee. All right. GI anthrax, we're not going to worry about. It's very rare, but you'd have to ingest the spores. Spores actually have a hard time causing infections in an adult GI tract, but so anyways, GI we're not gonna concern ourselves with. So treatment for this, very interesting, used to be penicillin, but after 9/11, when we had these cases in the U.S., the bacteria, the spores that were used, had a manipulated gene to resistance for penicillin, which was the drug of choice. So that totally changed treatment to Cipro. Prevention, we don't have prevention for the majority of the population, but military personnel do get vaccinated. So that's going to be one to, just to kind of keep in the back of your mind. It's not one of the high yield vaccines, but you should in general know that we do have a vaccine for anthrax. Okay. Real quick, just through Bacillus cereus, again, you guys can write this vignette. You pray you get a question on this because you're ready for it. But this is an intoxication. We get it from eating food that's stored warm but not hot, like fried rice on a Chinese buffet. And there's two toxins, but the one they almost always are interested in is the emetic toxin because it's fast, right? It's an intoxication. We're ingesting that toxin. And again, that's the one associated with fried rice, so we're not treating this. There's also a diarrheal toxin, cyclic AMP-inducing toxin, but again, nobody ever asked about that one. All right? So this is the one that's going to cause vomiting, diarrhea from fried rice very quickly, just like Staph aureus, almost exactly the same, non-bloody and with vomiting. All right. That's it. You guys did outstanding tonight. We finished right at 10 o'clock on the dot. It went fast for me, too. You said it went fast? God, it went fast for me, too. So we must have had a good time. That's a good sign if it goes fast and we're not like, ah! Okay. All right, guys. I'm going to let you go to your lives now. All right? And I'm going to go to mine. And we will see you tomorrow, right? Sunday night, we have a date. I'll see you then. Email me if you have, I said I put my email address. I'll just put it in the chat and you can go back and look. K Moscatello, you probably have this somewhere. Easiest one is gmail.com, right? All right, guys. I'll chat at you later.